attend the sessions you feel speak to you, as well as those sessions that you might be interested in, and spend some time reflecting on the talks. When I look at the program, I'm encouraged to see there is a lot of our 11 research themes reflected. And so over the last 10 years or so, we've had to put in a lot of effort to encourage more research from some of those understudied themes. A lot of our research was in the ecosystem structure, function, and process theme. And in about 2018, we decided we'd get together and we'd do a bit of a horizon scan on some of the major issues that are going to sort of face us in terms of managing the savannas and managing Kruger. And we found that these were some of the key themes. And so when I look at the program, and if you look through as well, you'll see either directly or indirectly, each theme is reflected in the program. And we are very encouraged by this because as I stand here today and another flood event has impacted on the infrastructure of Kruger, and we're thinking about how we're going to be rebuilding this infrastructure and planning for the future, themes like climate change, varying climate patterns, sustainable conservation financing, effective environmental management, and park planning has never been more real. So understanding these especially in terms of funding, often we never think of conservation as a business, but more and more we're starting to realize that conservation in Kruger at least is at least 2/3 funded by tourism. So understanding our visitors, understanding their preferences and future preferences, and staying ahead of the curve really is a conservation problem. So we're also encouraged to see that some of that is also making it into the program of late. Over the years, we've tried to enable scientists to do good work here, and I think if you look at the program, you start to see that there's a lot of great work happening in the park. But scientists of today have a greater challenge, and this is they function in a time of information overload, where the amount of data generated in Twitter, I heard this little info bit that the amount of data generated in Twitter in 2 years is more than all the books that have ever been published. So how do you compete in that space when social media influencers, they're 500 million and growing, are having a huge impact on what is truth? So you're finding ways and interesting ways to get your science out and make your story compelling is every bit as important as doing the science. And then an important sort of way of influencing is also to reflect with managers. And I think that that's what this meeting offers. When we did a survey a few years ago, a lot of you said that the reason you like working in Kruger, one of the big reasons is because you feel that your research has value and is taken up by management. And I think that that is really what we like to have this meeting do. So we invite managers as well to attend sessions that they find interesting, but also the socials. And so grab a manager and talk to them, reflect with them a little bit on some of the research. Reflective learning is often an underappreciated way of learning. Especially in terms of organizational learning. To take it from individual-based learning to organizational learning, we actually do need to reflect together. And so that's why we provided so much spare time in between sessions for you to do that. So with that, I'd like to officially open this year's meeting and encourage you to continue to discuss, debate, reflect, have robust discussions and disagreements on the themes covered in this conference, during this conference, as well as you go about the rest of your year. We encourage you to have some seeds planted and take up new research areas, so that it can have good value to our work of conservation in the park and in the global savannas. Thank you so much. With that, the meeting's open.
Thanks, Danny. So before we start the formal talks, I just need to do a couple of housekeeping arrangements. So just to remind us, this is the 20th, so we're having some celebrations. You will see that there are a few events that we've changed and that we've added to the program that we don't normally have. So yes, so we just like everybody to enjoy our 20th Savanna Science Network meeting with us, and hopefully we can build on it to go from strength to strength. So just a couple of announcements. So with regards to the formal presentation, so you'll see that we have a camera up front here, so all of it is being streamed. So please, if you do not want your talk live streamed, can you speak to Jackie about it in advance of your session? Because it means we will have to move you to the end of that session so that we don't stream that. So also, if I can just ask the speakers to please come to the front in the talk before yours, so that you can get set up if you don't want to use the handheld mic that I am actually holding. So just a reminder, our timekeepers are in the front here. Nikisha, can you just wave, please? So thanks to the team that will be doing the timekeeping. So she has just timekeeping notices that she will show. So she will show you how much time you have left in your talk. So please, guys, we have a very packed program. So I ask everybody if you can please just respect all the other people and stick to time. If you do use all of your time, the chairperson will actually get up and ask you to please stop your talk. So let's try not have that happen. If you do have space left in your time slot, we will then do some questions. So, and please also make sure that you load your talk before your slot. So the AV guys are doing that for us. So during a tea break, a lunch break, after the session or before the session, please bring your talks forward. Then a pointer is provided. Okay, so here's the pointer. Your talk will be on the bottom screen as well as behind you there. So it's really quite simple. Next, back, and in the middle over there, the pointer. So you're welcome to use that. Then any announcements or any changes to the programs will be announced in the mornings. So this afternoon we have one change. The patient's Shito talk has been cancelled. So the talk after that will just be moved up. There's also water on the table here on the right-hand side. So if you are coming up to give a talk and you haven't got your water, please feel free to bring that up with you. And then, of course, the horrible load shedding that, for those of you who don't experience it, you are very lucky. So unfortunately, Eskom, who provides us our electricity in South Africa, is implementing load shedding currently, which means that our electricity goes off for 2 or more hour breaks at a time. So we do have a generator which powers up the whole of Skukuza. However, it's not an automatic switch over. So when the electricity does go off, the electricity will be going off for about up 2 to 5 minutes. So if you are the speaker, sorry, we do apologize for that. But we will then just stop the clock, and once we have everything up and running again, you will be able to proceed from where you left off. So we're not going to be penalizing you for that. I know it's nobody's fault. So going back then to the meals and teas, the teas will be in the foyer, so you were given cups or mugs in your bag, so please bring those with you. The lunches will be served at the Safari Lodge, so that's where we had our meet and greet last night. Please remember your name tags, and the adjoining people or partners are welcome to join us for those. They have also been given name tags. So then as far as supper goes, Monday, Tuesday, and Thursday night meals will be served at the cricket club. So transport for this event has been arranged. The buses will leave here from the front of the conference center at 6 o'clock. So please make sure that you are here waiting in the parking lot so that you can be collected to get taken to the cricket club. They will then also be returning from 9:15 from the cricket club. There's only very limited parking, and that needs to be used by the conference organizer as well as the event organizers. So please make sure you use the bus transport that is being provided. So on Wednesday afternoon, there's a poster session that will be happening directly after the formal session, and we will then, later on, we will then let you know about the arrangements for Wednesday night. So just on the cricket club, please be aware that it's not a fenced-in area, so we will be providing lights and additional lights for in the case of load shedding. But please stay in the lit area, and please make sure that you don't feed any animal that may enter the area. And then there's water on the side tables on the side there, also, as I mentioned, for the speakers. But please feel free to drink the tap water. So this water is not from Gauteng, it's from the Sabi River, so it's clean and drinkable. So please go ahead and do that. You will also notice that we've added some additional conference activities. They're on your program. So I'd just like to bring your attention that this afternoon there will be 2 activities. So the one is the medicinal plant activity, and the other one is the herbarium visit. So please make sure to get yourselves to those, so we don't have arranged transport. We will also be putting up lists in the foyer. Unfortunately, we can only accommodate a very limited number of delegates, so please make sure you put your name up if you're interested. And then in addition, there will be some bird walks as well. So then there's a notice board in the foyer where we'll put up some notices. We also have a suggestion box with Jackie that you can make suggestions. Please, any outstanding payments, if Jackie can get those. We encourage you all, or the younger ones, I mean, I don't, so, but please feel free to use the Twitter account, hashtag SSNM20 for that. We're also moving a little bit with the time. So later today or by tomorrow morning, we're going to have a selfie frame that we're going to be putting up in the foyer. So I encourage you to get your group or get your friends and go and have selfies taken. Also, if you've been a group that have been coming for a lot of years, have a selfie, and then you can also just upload those onto the Twitter account. Then the AM Spa, not only did they give us two lucky draws for last night, but you'll see they are also giving us a 12% discount on any bookings over 250 Rand. So if you or somebody that you brought with you would like to make use of that, the promotion code is SSNM2023. Very easy. If you forget it and you want to make use of it, just ask us about that. So I think that's pretty much all from my side. We will be making arrangements or discussions at certain points in the program, but those are all the formal announcements. To those that haven't been at network meetings before, we're really happy to see you. We look forward to networking with you. If you do have any questions, you're welcome to ask. Any of us that you can see are busy with the organizing, so some of the timekeepers, myself, Shanae, who's at the back there, or Jackie, if you have any other queries. So we're now going to start the formal presentations, or the formal part of the meeting. So I'm going to hand over to our first speaker, who's Nikki, and she's going to be telling us about grassy ecosystems in the Anthropocene. Thanks, Nikki.
Hi, everyone. It's very nice to be back here. So today I'm going to give you a very broad overview about grassy ecosystems in the Anthropocene. I was asked to review this once and to talk about how grassy ecosystems are being impacted in the Anthropocene with this ever-increasing human pressure on them. The first brief was to talk about all the grassy ecosystems in the whole world, which is very daunting, and I'm not that well-traveled that I've seen all the grassy ecosystems in the world, but it was quite fun to take a perspective of an African ecologist looking outwards. I decided to try and limit my extent a little bit and decided to focus on grassy ecosystems in the Anthropocene that occur within the ecosystem uncertain space. So basically ignore what's happening in edaphic grasslands, but focus on the drivers of change in many of these grassy ecosystems, which are disturbance-driven, consumer-controlled systems which occur within a climate space where forests can also grow. So I have written this extensive review, and you can read all about it, and I'm not going to bore you with the fundamental details because there's a lot to say. But I'm going to take this approach of highlighting some of the lessons I learned by sitting there and taking this huge zooming out view of the world's grassy ecosystems. I'm going to talk about 4 lessons that I've encountered that I think are actually really important as a grassland or grassy ecosystem community to bring to solve and try and get some of these issues on the table. So the first challenge I experienced when trying to understand what the grassy ecosystems are, what's happening to them in the Anthropocene, is we actually don't know where they are. And this is actually a really fundamental problem, because how do you conserve or how do you protect or take advantage or manage of a system where you don't really know its actual distribution? This is a map from 2014 which maps the world's grassy ecosystems, and this has basically taken the terrestrial ecoregions and mapped them where there's at least 10% total area of grassy ecosystems. So it basically ignores most of the Myanmar, the Southeast Asian areas, but this is what it reckons where grassy ecosystems occur. If you look at the WWF ecoregions, here I've pulled out temperate grassy ecosystems and tropical grassy ecosystems, and this map suggests that there's far more grassy ecosystems than the previous one, which is really nice, but it still excludes fairly large grassy ecosystems or potential grassy ecosystems in Asia. Then there's this very nice paper that's in preprint, which highlights, I think there are quite a few authors of this paper in the audience, which instead of mapping the actual distribution of the biome, mapped where grasses actually occur. And this map shows that the potential grassy space is much, much broader, and there are many more grassy ecosystems in the world. So this brings us to the next problem. If you see a grassy ecosystem, is it actually a true grassy ecosystem biome, or is it just a product of deforestation? Which brings us to this next lesson that I learned, and it's not new either, but it was very profound to see how extensive this problem is this persistent misclassification of grassy ecosystems and the cultural attitudes assigned to them. This has really come out with the sort of Humboldt's science and exploration of the world, and during the time of colonial expansion, a lot of travelers were moving around the world, and they came up with the idea that if you understood what the climate and the soil was for any part of the world, you would understand what the biome is, with the idea that vegetation is really predictable by climate and soil, which is true for many parts of the world, but this does ignore the fact of consumer-controlled systems. This also gave rise in this sort of spread of the global ecology and this focus on succession theory, which sort of notes that forests are the natural climate space, natural climax state, and if you're a non-forested system, you either non-forested because you're on the way to becoming a forest, or you are in a state of arrested succession where you are experiencing degradation. So this is a very pervasive issue across many of the grassy ecosystems. And many people, again, in this audience have actually highlighted this, and it's a very common understanding that we have this problem for tropical grassy ecosystems. But what really astounded me was actually how extensive this narrative is across many of the grassy ecosystems of the world. And one thing that keeps coming up to me that actually is quite a fundamental problem is this narrative of desertification. A lot of this arose in sort of the French colonies in West Africa when they arrived in these drylands, coming from very lush, moist, forested landscapes. They saw these dry areas which are very prone to drought and wet, dry cycles. And so this is some of the texts from these areas which sort of say they saw these deserts and droughts as results of divine punishment. And looking at these systems that seem so dry and desolate, they viewed them as really, really degraded and blamed the locals for cutting down all these trees and destroying the forests and the landscape. And the incredible ecosystem resilience post-drought was really largely ignored. And then these texts also note that they blame this desertification on clearing land for cultivation, fires, grazing and browsing, and cutting trees for wood use, which meant that the management response to this was to say stop using fire and herbivory, changing the way people use the landscape to help restore this landscape and make it more productive, and sort of using it for other purposes. But also there's a very nice, interesting legacy. If you travel around Africa, you can go to many savanna areas and you inevitably bump into a forest reserve. And these also have this long colonial history where these areas were deemed as degraded and deforested and these fences put around and fire and herbivory being excluded from these landscapes. And of course, this has given rise to this huge push of, in current times, of this fight against desertification and led rise to very well-funded things with the African Great Green Wall, with the aim that you want to plant trees in these arid systems to arrest the expansion of deserts. And in China, there's the Great Green Wall there, where large amounts of trees have been planted to regreen the desert and help prevent its expansion. And I think the ultimate aim there is to plant a massive green wall all the way from Africa all the way through to China. But that is really an important narrative, and it has a lot of power. What also was really interesting is, and we know that grassy ecosystems in the tropics are often misclassified, but it's astounding how widespread this is in temperate grassy systems. And a really nice example comes from the Russian steppes, where in the 1700s, a massive action to reforest these landscapes was initiated. And German foresters were encouraged to come to Russia and set up forestry and set up schools to teach how people to re-establish their forests in these Russian steppes with the aim of increasing water available by increasing rainfall by planting so many trees. And there's this persistent issue, which I found really interesting now living in Europe, where I haven't before, was this discussion about semi-natural grasslands in the European context. Semi-natural grasslands everyone talks about frequently, and they are basically defined as remnants of habitats created by low-intensity traditional farming, with the idea that, well, the recognition that they're very biodiversity rich, but that they're cultural landscapes from previously deforested areas, and now they are open areas maintained by humans, and they're viewed as a very anthropogenic area. And there's some really nice research, I'll just quickly talk you through this, about some of these semi-natural grassy ecosystems in Europe, with the suggestion that actually consumer-controlled grasslands are actually very common. There's a high proportion of them in the landscape in the early Holocene, where the suggestion is warmer and drier temperatures and this megafauna in these landscapes kept these grasslands, these primary ancient grasslands open. Then, with the mid-Holocene, when the temperatures got a bit cooler, and there was the extinction of megaherbivores, low-intensity human impact sort of replaced the role of megaherbivores and maintained these systems and kept them open. And only really in more recent times, 2,000 years ago, is there really a clear impact of deforestation increasing the grassy area. And arguably, you could say the grasses that fall within this triangle are semi-natural grasslands, and actually there's potentially much more ancient grasslands in Europe. Anyway, that's a debate I don't know really much more about, but it's a very interesting topic. So, aside from this persistent misclassification and lack of understanding of exactly what the true extent of our grassy ecosystems are, there's this other very interesting lesson which I was actually really surprised about how extensive this problem is the conversion of grassy ecosystems. And I want to start off here by saying we're probably underestimating this extent because we actually don't know where our grassy ecosystems are, largely. And there's some areas which have really been extensively cleared. They're often in the northern hemisphere. There's some estimates that European grasslands, there's only 2 to 3% of their original extent remain. Whether this includes semi-natural grasslands or not, that's sort of something that needs to be dealt with. Okay, I'm going to carry on. So, I was talking about the conversion of grassy ecosystems and that it's extensive. There's been extensive conversion in the Russian steppes. These conversion waves are often promoted, often occur very rapidly. The Russian Virgin Lands campaign meant 460,000 square kilometers of land was converted very rapidly. And estimates there's only about 20% of these areas left. Similarly, in the North American Great Plains, there was a huge wave of conversion, and it's estimated about 40 to 50% of the original area has been converted, with a very rapid rate of new crops being established in primary grasslands. But the good news about these is that the rate of conversion is declining. The bad news is the rate of conversion is accelerating in the tropics. And since 2003, 17% of the global land agricultural area has been planted, with most of this occurring in Africa and in South America. And the Cerrado is having a really tough time. Technological developments in the 1990s to reduce soil acidity has meant that there's been a rapid conversion wave, with estimates about 50% of the native vegetation being lost. And then very notable in the Cerrado is the current rate of change is about 1% per year, which is very rapid, and the rate of conversion is much bigger than that of the Amazon, with a lot of the activities, avoided deforestation activities, spilling over into the Cerrado. And in Africa, a lot of non-converted land remains, but since 2003, 79% of the new land that's been planted is being planted in non-transformed vegetation. And here the hotspots of expansion are in the Sahel, West Africa, and the Rift Valley in East Africa. So just a reminder, there's an emerging field of research which really is highlighting that once you cross this threshold of cultivated land or converted land, it's very difficult to reverse the conversion. And so some suggestions or modeling studies suggest that it takes as much as 1,000 years for these grassy ecosystems to recover, if at all, unlike forests which sort of seem to recover fairly quickly. And so it's really important that we should be really managing this and being very aware that the cost of converting these lands is fairly permanent. So what's happening to the non-transformed grassy ecosystems? Of course, there are a lot of grassy ecosystems that aren't transformed. They may be utilized, but they haven't actually been plowed up. And there's some very interesting trends that are happening across the world's biome, that there is this persistent shift of plant functional types across the world. This is a map of all sort of meta-analysis of all the studies which have documented clear changes in plant functional types relative in natural or semi-natural ecosystems where the change can be attributed to land use or climate change. So just to summarize what's happening in the grassy ecosystems, there are three very clear trends. There's a clear trend of forest encroachment into these grassy systems. This is very clear in the tropics. There's been quite an emerging amount of research showing that there's a lot of expansion of forests in the South America, Africa, and Southeast Asia into these grassy ecosystems. But this is a very prominent problem in temperate systems where there's a clear spread of temperate forest species into grassy biome. Something I've spoken about really a lot over the years, I'm not going to do it more, is that there's woody encroachment within these grassy ecosystems. So these grassy ecosystems are becoming much woodier, not only in the tropics, but also in the temperate systems. And there's a range of studies which sort of highlight how extensive this is across these different landscapes. And the third trend, which is, I would say, emerging, but still has a lot of work to be done, is there seems to be this increasing trend of expansion of grasses into deserts and arid shrublands. This is increasingly noted, but it's not widespread. We can sort of say this with low confidence, but it seems to be an emerging trend that you are seeing these grasslands expanding. And it's quite clear, there's been very clear studies in the Karoo, the Sahel, in some Asian deserts, and also in the USA. Often this expansion is associated with the spread of invasive grasses, and this has an important ecosystem consequence. It has benefits in that it brings grazing to previously ungrazable land, but it does mean that there's fires moving into areas that never burned before. And the drivers of these are really, I think the take-home message from this, and I'm not going to talk more about it, is the drivers vary, and the strength of our drivers vary. So what's happening in one system with fire suppression in one system may be causing this, whereas climate change in the other may also be causing this. But this is sort of anecdotal evidence, and there isn't a sort of a global framework which helps us. We have very little predictive power as to understand why these changes are occurring at a global scale, and something we should probably strive to achieve a bit better. And I think I'm going to finish there with some final thoughts, and sort of highlighting some of the really big gaps that I noticed. There are some very notable gaps. One that struck me quite clearly was that we're actually really bad at understanding the impact of climate change in these tropical grassy ecosystems specifically. It's probably better we have a better understanding in the temperate systems, but we really haven't got to grips with this. Some of the problems is that these grassy systems are fundamentally linked to land use, so it's very difficult to separate the role of land use and climate change. And there's been some work emerging from this. I hope Steve Higgins will talk about that. But it's quite difficult to attribute what the role of climate change is. And yeah, so then the second point is the secondary grasslands. We don't know how to restore them. And I think the final point that I just wanted to make, as a community, we should be making more of a case of why this matters, and we should be really seeking to answer what are the consequences of losing these grassy ecosystems and highlight their importance into the future. And I'll stop there.
Thanks, Nicola. You've used up all your time, so unfortunately people will have to grab you at your lunch. So I'm the next speaker. So for those of you who don't know me, I'm Judith Buerta, and I work here, and I have worked here in scientific services for a number of years. So today, me and my co-authors, who I'd already like to thank up front, Isak and Louise, who have spent many meetings here with me and are going to spend hopefully a lot more meetings, put this presentation together. And we just like to reflect on the last 20 years of the Savanna Science Network meeting. So it's going to be broken up into two components. So the first part of it is going to be looking at the delegates and the representation and who the affiliations are, etc., and how that has changed and grown over time. And then the second part of the presentation is going to be focusing on the abstracts and looking at can we say that we are making a positive contribution to biodiversity with the abstracts that are presented here at the meeting. So to start off with, as Danny mentioned, these three gentlemen on the screen are the founding fathers, as we call it, of the Savanna Science Networking meeting. So William Robertson, or Bill, as we know him, was in the early 2000s, he was funding, or the Mellon Foundation was funding the River Savanna Boundary Program, and Bill was the project manager and very involved in Kruger. Harry Biggs was from Scientific Services, and he was the PI from our side, and then Kevin Rogers was the PI from the program from WITS. And after the 2000 floods, they were sitting around a table discussing ideas of how they could have meetings where river-related issues could be discussed. So the meeting started off very small, as between 50 and 30 delegates, the first couple of meetings. Everything was done in-house, so Wendy Midgley from WITS did the catering, and only river-related issues were discussed. The meeting at that point was fully funded by the Andrew W. Mellon Foundation. So then it started taking off, and the meeting started expanding, both in the number of delegates that were present, but also broadening to look at Savanna issues or Savanna research based mostly work from Kruger, but then also broadening in focus and looking at savannas or international savannas, and it then morphed into the meeting as we currently know it today, being the International Savanna Science Networking meeting. So when we look at our delegates, or the number of delegates who have been attending since 2008 to 2023. So all the slides that I'm going to be presenting up until I look at the abstracts, look at the information from 2008 to 2023. The reason for this is that this is where our most accurate delegate information actually resides. So from the graph, we can see that we had 1,321 unique delegates. So basically, those were the people that, the unique people that attended the meeting. You can see that a lot of this, the majority, attend one or two meetings, but we do have some real moral supporters. So you'll see that 117, or 8.9% of all delegates attend more than 5 meetings. So just remember, if you're thinking about how many meetings you've attended and where you fit into this graph, we are only looking at 2008 until 2023, and I know a lot of you were involved with the meetings prior to that. So then we look at the institutions that the delegates have come from. So in total, we've had 341 institutions that have been represented at this meeting. So you'll see it started off in 2008 with about 50 represented or affiliations. This then increased and it started stabilizing. And then, of course, we all know the dreaded 2020, this year here, that's when COVID hit us. So we were lucky to get a meeting in just before COVID hit. I think the first COVID actual people that the first COVID was happening, we were sitting in this room and we were hearing about it and thinking, oh no, this can't affect us. And it did. We didn't have a meeting in that year. The meeting after COVID, very low representation. A lot of the overseas universities and students couldn't travel. And then, of course, we're up here again with 2023. So on the right-hand side there, we're just looking at the organizations that have more than 20 delegates attending over time. So at the top, so all the shaded ones are the South African institutes, with the white ones international representations. So you can see it starts off, Sandparks is prominent, which we should be, and then a lot of universities, and then we start seeing that even the international Wageningen, the University of Jena, University of Flora are very prominent in the top part of this diagram. Then again, we get a lot of some South African, and then we get a lot of the overseas institutes. So you can see that there's been good representation from a lot of different organizations. So just then breaking this up further to try and see or how we categorize these institutions. So we can see that the majority of the delegates are coming from either university or conservation and research council institutes, with then the smaller amount of delegates being people like from the media and government and various other organizations. So then focusing on the conservation side of things, we can see that a concern for us is that this is the percentage delegates over time that come from conservation organizations. So be it sandparks or conservation meetings outside of sandparks. And the worrying fact is you can see that both of these delegate numbers are actually declining over time to where we are in 2023. So even the sandparks contingent, they were a lot in the past, which is also declining. So one needs to think about this. So what could this be? Could it perhaps be that conservation organizations are struggling and they don't have funds to attend these types of meetings? Or is it because the meeting is becoming too academic and not as applied as what the conservationists are interested? So just something to keep in mind as we go through the meeting this year. So then, as I mentioned, the international or the internationalization of the meeting started in the early years as well. So the left-hand graph shows us that 35 countries have been represented. It started off with a really low number of countries being represented. It then continued to climb, and this year we have the most countries present that we have had at a meeting. The right-hand graph just shows this very nicely too. We can see that at the beginning there were very few international delegates, mostly South African, and then by this point in time they were just converging on each other. And then again you can see the big COVID effect there, and then this meeting roughly 50-50. So 50% from local, 50% from international guests. So this is just the attendance of countries outside of South Africa. So this is just highlighting what country makes up the biggest proportion of the international delegates. So you see it's quite heavily dominated: USA, Germany, Netherlands, and the UK. Concerning on this slide is that Zimbabwe and Botswana, who are our conservation or have big conservation organizations in Africa, have hardly any representation at this meeting, and we'll see that only 6.8% of all these delegates are actually coming from Africa. So then we focused our thoughts a little bit about looking at the abstract, and the question that we asked ourselves is, does this research make a difference in saving biodiversity? Okay, so that's quite a hard task. So we've seen that over the 20 years, this meeting has really grown. It's grown in delegate number, it's grown in the number of organizations that have been represented, and it's grown in the number of topics and the themes that we have discussed over the years. But can we say that this meeting is actually making a difference and making a positive contribution to saving biodiversity? So how we thought of doing that was looking at this Williams framework, so the past and future role of conservation science in saving biodiversity. So they put together a framework whereby we can actually look and try and make sense of whether these abstracts and these presentations are having an impact, of a positive impact towards biodiversity conservation. So what Williams did is they put together a framework for how effective conservation research could progress. So it looks at identifying problems to diagnosing their proximate and ultimate causes, and from proposing to designing, implementing, and testing the responses. Okay, so that's quite a mouthful. So this is really what the Williams framework proposes. So you can see in the top side of the graph, there's something called research on threats, and at the bottom, they look at research on responses. So really, the top part of this is we start by describing the state of the conservation target, then we understand the mechanism which causes an unfavorable state. We establish the source of the threatening mechanism, and then lastly, we identify the underlying driver. Okay? Then at the bottom of the graph, we look at the research that happens on the responses. So we're interested in proposing a response to the threatened mechanism designing it, implementing, and testing this response. Okay, so pretty straightforward. So how we've done it in the couple of slides that are following, we've divided our stuff into red, which is patterns and threats, and the responses at the bottom. And those 4: state, mechanism, source, and driver. So that's what I've just explained. And the proposed, design, implement, and test is the 4 subcategories on the bottom. Okay, so what Williams did is that he applied this framework to 959 conservation journals over 20 consecutive years from 20 different conservation journals. And the alarming factor is that he found that 43% of the research is focusing on the state of biodiversity only. The linking of mechanism to the source driver was only 10%. So are we then thinking that the conservationist scientists are not developing a deeper understanding of the threats? 70% of the work had no proposed response, and there were very few studies that looked at designing and implementing change. So we then took this a step forward, and we applied the Williams framework to the Savanna Science Network meeting presentations. So because of the amount of abstracts that we had to analyze, we only took 3 time steps, so namely 2004, 2013, and 2023. So we then went and we categorized them into what type are they? Are they ecological, social ecological? And we then divided them into these 2 different main categories, as I mentioned, so red being patterns and threats, and the responses in blue. And then, of course, the 4 subcategories under each of those. And what did we find? Okay, so this is just an example of how we went about it. So you'll see all the patterns and threats are some of the abstracts from this meetings that fall into the different categories. So you'll see a nice one that would fall under a driver would be the one about fire influences and biodiversity by modifying vegetation structure. And then we looked at the work, the responses, and a good test response abstract was dehorning effects, male white rhino territory, size and location in Itala Game Reserve. So a similar approach was done for all of the abstracts for those 3 years that I mentioned. So what do we see? We see that, so this is supposed to be red, but it's a bit orange, but that's okay, you'll still get it. So really, the threats are in red and the responses are in blue, and we can see that a lot of our work, or the majority of our work, is actually falling into the threat category. So if we look at the table on the right, or this little part on the right-hand side, so Williams found that in his study, there was 30% that fell into the threat category, and in the responses category, and we can see that we only have 20.4% that's in the response category. So we are still largely in the threat and patterns category and not yet in the response category. The SORMA meeting, for those of you who don't know, is the South African Wildlife Management Association, and they found that up to 41% of their research was focused in the response category, and this could be because it's more applied and a lot more conservation organizations attend this meeting. So then we quickly went and we looked at the subcategories within that, and we can see that if we look at our patterns and threats ones at the top, we can see that the research around drivers is definitely has been increasing over time. We're up to 43%. And then if we looked at the test, this is very light, this blue line at the bottom is the test response. We can see that that's also picking up, which is great. Okay, then just quickly, we can see it's dominated by ecological research. 2013 and 2023, you can see that the social and the socio-ecological research has been picking up. If we look at the presentations that were addressing the responses, you can see the ecological side, there's only 11%, whereas the social and social ecological is a lot more. So it could be that they came in a bit later, or that their research is more response-driven. It's just something else to keep in mind. So I'm almost done. I know my time's running out, but just a couple of last words from my side. So we can see that this meeting is highly diverse and has a dynamic delegation attended, and it has grown both in format and in size. There is a concern about the number of delegates that are actually coming from South Africa, and the conservation organizations. So when we focus on whether these abstracts are positively contributing to the biodiversity, the reality is that exploring responses is complex and difficult to research. However, ecology is what we do, and we have had wonderful contributions to science over the past 2 decades at the Savanna Science Network meeting, and we celebrate this today and this week. So I'd just like to ask that people keep this caption in their mind when we go through the week's proceedings. So it's something that Michael Sewell said in 1986. If conservation biology or conservation science becomes isolated in the mental world of academia, it will be of little use. Its prescriptions will not be informed by the real-world problems of the people who are most involved and affected. Okay, so I'm not quite done yet. I know I'm over my time, but I just need half a minute to complete my thanks and to just give 2 gifts out, so please bear with me. So a meeting like this definitely won't be as successful as it is. I think we can all attest to that. So I'd just like to thank our sponsors that we've had over the years. So as I mentioned, the A.W. Mellon Foundation started, and then SAON or NRF, Global Supplies, and Jackie with Mpumalanga Promotions, they've supported us over these years. So thank you very much to our sponsors. We really do appreciate it. Then behind the scenes, guys, this is a lot of work. You know, I'm not sure if you've never organized an event like this, you don't realize what happens and what goes on behind the scene. So just to the dedicated task, you're not all in there. I'm sorry about that. We've had people coming and going over the years. So thank you very much for all of your help. You're always there when we need it. And then I think, as the saying goes, it takes a village. And definitely, with the Savanna Science Network meeting, this is completely true. So last slide. So these are our premier members. So I'd just like to thank the delegates, those who keep coming back. So the ones in green are all of those delegates who are actually attending this meeting. The ones in the hashed green are the scientific services or the Sandparks delegates. So this is everybody that has been here for 10 or more times. So I'd like to just take this opportunity to thank our best attending non-Sandparks delegates. So I'd like Francie, and then our best attending non-South African delegate, Greg, just to come up to the podium quickly, and I'd like to present you with a small gift. Thank you. Can you hear me now? Perfect. Great. Thank you. So while we wait for the electricity to come back on, I just want to introduce the next session, which is about invertebrates with a special focus on termites as ecosystem engineers. And I'd like our first speaker, Paul, to come closer so that we can get you set up so that you're ready to go when the electricity comes on. Thanks.
We'll appear in a minute. They're there, but this isn't the first time I've had to deal with these kind of power cuts. I once gave a talk where the slide projector entirely failed, so I had to give a talk without slides. And then on a very memorable occasion, the power went off entirely in the middle of the night, so I had to give a talk in total pitch blackness. So I am quite used to one or two technical problems. There you go. Right, let me introduce you to the big six. You've heard of the big five, of course, because there they are, the trophy hunter's dreams, with a camera, hopefully. But I want to introduce you to the big six. The other player in this is termites. Now, macrotermes, which you can see here, is found throughout Kruger Park. It's a massive, impressive structure with millions of inhabitants, and it also has a great advantage in that it doesn't move. So you can see it very, very easily. So I suggest termites should be educationally one of the things that people consider when they are thinking about the savannas. So there are some termites doing their stuff, and that's the mound they built. Now put your hand up if you've ever seen a mound and wonder what it was. Nobody? You all know what they are, sorry. Yeah. So this talk, which is going to be the introductory talk to a whole series of talks on termites, is going to be dealing with termites in African savannas. And although I've put my name first, a lot of this work really belongs to a lot of other people, particularly Andrew Davis and Kate Parr, and a lot of what I talk about is going to be related to that. So, in savannas across all continents and across all biomes, across all continents, you'll see these mound fields of termite mounds, which are very prominent. This is one from Ghana, from the north of Ghana. And one of the striking things about these mounds is that they are often multi-inhabited. So often you'll find grass feeders at the top, soil feeders in the middle, and fungus-growing termites at the base. So if you include the macrotermes and all of the grass-feeding and soil-feeding termites, there's a huge biomass of termites in savannas. So what are termites? They are eusocial cockroaches. Now this comes as a big surprise to many people, but they are in fact most closely related to cockroaches and not to ants, even though people have referred to them as white ants. They are not ants at all. They're not closely related to them. In fact, their common ancestor was 400 million years ago. All species are colonial. All of them are social. All of them have a colony center. There are, among termites, two sorts of eusociality. There's primitive eusociality, which is wood-nesting termites that nest and feed in dead wood. They have no true workers, and they practice fortress defense. So they are often found in dead wood, where they occupy it fairly simply, and these are primitively eusocial. Although they have soldiers, the soldiers are not otherwise very complicated, and they do not have complicated lifestyles. However, most of the termites that we see are foraging termites. These are termites with true workers that forage, build, and tend immatures, and they are fully eusocial. Now, 99% of all African termites are foragers. So most of what you find in, say, the Kruger National Park will be foraging termites. Most species have soldiers, and these are a specialized caste that only defend the colony and the foraging termites. So they send out foraging termites to collect food, and they are defended by the soldier caste. Now, foragers feed on a range of materials, on wood, on grass, on leaf litter, plant debris, humus, and soil. So they have an important role throughout in recycling a dead material. Generally, except for grass, they feed on dead organic material, plant material. So here is a termite mound. The colony is inside this structure, inside the mound, and you can see this gigantic queen in the center of the colony. There's also a king. So unlike Hymenoptera, they have both a king and a queen. And the king and the queen mate continuously throughout the life of the colony. Now, the colony can live for 50 to 70 years, and often the same male and female reproductives are present throughout that time. They also have these workers, which are generally major and minor workers. Oh, could somebody bring me some water? Thanks. Thanks a lot. It's around a bit dry. Now, the workers do all of the jobs in the colony. They forage, they do the man building, they feed the young and each other, and so they are really the main body of the working groups in the colony. The soldiers, of which there are two types, major and minor. Oh, sorry, where's the minor? Major and minor. Only job is to protect the colony one way or another. Now, they generally are not very good at doing this. All they really can do is protect the colony long enough for the workers to reseal the breaches that may have been made in it. And we think that there are two sorts of workers two sorts of soldiers, because the major soldiers are to deal with tumatophagous mammals, and the minor soldiers are to deal with ants. And throughout this story, you'll find that ants are vitally important as predators of termites. So, what are termites like in Africa? They are the most diverse continent, by far. There are many more genera and species in Africa than there are elsewhere. The highest biomass of termites is also in Africa. Oh. Thank you. Sorry, I'm going to disappear from view now. The most diverse continent is Africa, and African rainforests have by far the most. They have the highest biomass of in the soil of any group in Africa. So African termites have a higher biomass than, say, South American or Australian or Asian termites. They're also the most diverse ecologically. There are more different kinds of termites that feed on different things in Africa than elsewhere. So I'd say overall, the ecological importance is greatest in Africa. So it's worth noting that this continent has by far the greatest diversity and ecological importance of termites. So the two things which are important for termites in Africa are soil-feeding termites, especially in rainforests where they can be incredibly abundant. We've measured something like 100 grams per meter squared of termites, soil-feeding termites in African rainforest. But the other thing that's vitally important for termites in Africa is fungus-growing termites. These are the macrotermitinae. These are termites that grow fungi in their nests. And these include the things like macrotermes, which produce these very large mounds. And these fungus growers seem to be extremely important in savanna ecosystems. So, it's also worth noting that the rest of the world's termites really are out of Africa. They have come, the higher termites, as they're called, the Termitidae, so this is the majority of tropical termites, evolved in Africa, and they have dispersed to other continents to a greater or lesser extent. Unfortunately, although we know a lot about termites in rainforests, we know relatively little about termites in savannas. So I'm going to do a very quick whistle-stop tour through recent studies on termites in savannas. Let me start by saying that termites have an enormous biomass in the world. So this is a paper that I co-wrote in 2021, where we had tried to estimate the biomass of the various groups. And as you can see, ants were estimated to have more than termites, but both of them together were by far greater than in any other terrestrial arthropods put together. So you'll notice that livestock is the largest biomass. But you can see how ants and termites are vitally important in numerical terms. Just this year, an estimate was made of the total number of invertebrates in soils. So these are biomass. These are biomass estimates for from this paper in Science Advances by Ron et al. And they discovered that termites were incredibly abundant in the soil, making up 40% of the biomass of invertebrates. So, where are termites found? This is a recent map of global diversity constructed by Joel Woon, who was a student that I shared with Kate Parr. And this shows that the colors are, the lighter the color, the lower the termite richness. So overall, north and south temperate regions don't have a lot of termites, but African rainforest is the center, and South America has many, many of these. This has been constructed from examining a lot of studies of diversity across the world. And so now this is, I think, the best estimate we have so far of global species diversity. We also, Joel also attempted to estimate biomass, which is slightly different from the species richness. And you can see that here there are various areas which differ from there. And you'll also see that biomass tends to creep more highly, to be higher in areas like South Africa, which don't have very high diversity. The other thing that Joel did was use the data on earthworms that already existed to show whether earthworms and termites were coexisting in the world. And as you can see, earthworms are really predominantly in the temperate region, and termites are in the tropics. So we're talking about the drillosphere, which is the top and bottom areas, and the termitosphere, which is the area in the middle. So, what is the context of termites in savannas? We can think about the fact that African termites mostly evolved in rainforest. And they colonized savannas once they appeared, which was probably in the Miocene, maybe 15 million years ago. So moving from rainforest to savanna was a major environmental change. So how did they do this? How have they made this change? Well, we can look at the forest to savanna transitions by looking at the number of genera. So there are 61 described genera in forests, and 32 of those have made the transition from forest to savanna. So some of the genera within forests have become adapted to live within savannas. And within that area, a certain number, 20-something, have actually evolved in savannas. So these are genera that are only found in savannas. And these are almost all grass-feeding termites. But how did they get into savannas in the first place? Well, this again is the work of Joel Woon, and he looked at the thermal maximum and minimum of various of these species in dark boxes are savannas, light boxes are termites, are rainforests. And as you can see, the dark boxes, the savannas, so those are critical minimums, and those are critical maximums. So as you can see, the range between critical maximum and critical minimum is smaller in the rainforests than it is in the savannas. So they do seem to be adapting and changing their thermal tolerances to cope with savanna systems. And this can be summarized. So on the x-axis here is the leaf area index, a measure of canopy cover. And here is the critical thermal maximum of an individual termite. So if you look at that, it's a very clear correlation between canopy cover and the thermal maximum. So as the canopy cover drops, the thermal maximum increases. So they are adapting to the changes in the environment by changing their thermal tolerances. Right, well, later on in this symposium, we're going to hear from Amy Zan, who's going to talk about her work, which was published last year in Science, looking at termites and decomposition. And this is mostly in rainforest sites. I'm only going to talk very briefly about this because I know she's going to talk in a lot more detail. But she has all of these sites throughout the world, some tropical, some temperate. And the take-home message from her work, which we'll hear all about later, is that the savannas have the greatest level of decomposition due to termites. So termites are better at wood feeding than microbes, which is what she's going to say, particularly in hot, dry regions. Amy will be giving a much more detailed description of this work later. The only add-on is that my postdoc, Stephanie Law, has looked across the woodblock data and looked at whether those woodblocks that have macrotomitiny, so those are the ones on the left, have a higher K value, have a higher decomposition rate than the other areas, some of which don't have fungus-growing termites, because as I said, fungus-growing termites are very much part of the African system. And she found that termite-mediated decay rates are significantly higher in regions where fungus-farming termites are present. So the presence of these fungus-farming termites is vital to our understanding of rates of decay, potentially, in ecosystems. Okay, now I talked a little bit before about how ants are voracious predators of termites. Well, we did an experiment which Alice Walker, a student again of Cape Pars, did, looking at whether if you used poisons to suppress ants, whether termites would do better. And so if you look at these diagrams, these are for wood, grass, and dung as the decomposition substrate. And these black lines are controls, so there, and there. And these dotted lines are places where ants were suppressed but termites weren't. And so on the x-axis is the days since litter bags were presented in ecosystems. And as you can see, for both the wood, grass, and the dung, decomposition was much greater when ants were suppressed, the dotted lines, than when they were not suppressed. So it looks like ants are really strongly controlling decomposition by termites in these systems. And if you look at these, if you look at the relative contribution of termites and microbes in these systems, you'll find that in the ant suppression, in each case, there's a much greater contribution of termites to decomposition when ants are excluded. We also looked, and this is another student who Kate Parr and I were involved with, Kath Bunny, looked at sites along a precipitation gradient, where we looked at how important termites were, and we looked at wood, dung, and grass. And as you can see, that as it gets drier, so the important thing to look at is the percentage due, sorry, the amount due to termites, which is the black bars. And as it got drier, the contribution of the termites increases throughout. So in drier, hotter areas, although these are potentially drier rather than hotter, in drier areas, termites become more important. Now we have expanded this to include sites from Ghana and Gabon, which are rainforest sites, and we've been able to look at decomposition rate, partitioning out microbes and termites using two sizes of mesh. And we were able to show that as the rainfall drops, the absolute termite decomposition proportion, so the absolute amount of termite decomposition also drops. And for a lot of these drier sites, termites are responsible for more than half of the decomposition in the system, so they're really important. So it's really that plot that shows that most clearly, annual rainfall on the x-axis and the amount of termite decomposition on the y. So overall, it's telling a story which we've known about for a long time, that as it becomes drier and more seasonal, termites become more important, and then even if their value, if the amount that they contribute drops, so the absolute value may drop. Termites as decomposers become proportionally more important. And you can see this particularly in very dry desert ecosystems like those in Australia, and that Baptiste is going to be talking about later, and those in North America. Finally, we have to think about the fact that termites can help to increase the fertility of areas which are dry. So this is Kenya, where each one of these is an odontotermes mound. Now, the surrounding area is not suitable for vegetation. Vegetation can't be grown. But in areas where the termites have brought all of the nutrients together, have concentrated them, so as you can see here with these round areas, they are areas which can sustain vegetation. So although overall, if you were to simply mix all of this soil up together, it could not sustain vegetation. Overall, it can because of the termites. So, what do we conclude? That African termites mostly evolved in rainforest. When savannas became abundant in the Miocene, some termites occupied them. Apologies for the mess up of the formatting here. In many ways, savannas are good areas for termites because termites can control their environment in a way that other invertebrates can't. So they can often prosper when other organisms cannot. Especially see this in decomposition in drier savannas, as I've said. For more details, stay in this seminar. Thank you.
Thanks, Paul. Your time is up, but thanks for setting the scene for our invertebrate talks to continue. So then next up is going to be Baptiste, talking about termites and decomposition. Thank you.
All right, so I'm going to talk to you today about the spatial variability and the contribution of termites to decomposition. So as Paul has just mentioned, termites are really important mediators of the carbon cycle. They've developed this symbiosis with bacteria, which allows them to consume recalcitrant vegetation in the ecosystems. And they've adapted especially well to dry ecosystems and dry and warm ecosystems. So this study by Byron et al. This year has shown that about 40% of all soil arthropods are termites, showing their omnipresence in these systems. They're especially important in savannas. And also they've been shown to be islands of fertility, as Paul has just shown with these pictures of drought during savannas. And they've been shown to consume up to 60% of all deadwood in savannas, showing their real importance in these systems. But however, they're pretty heterogeneous throughout the landscape. So as we can see in this study by Lavelle et al., we can see that there's shifts in abundance across biomes. So if we focus on the savannas here, the termites are the light green bar, we can see that there are about double the density of termites in these savanna ecosystems as there are in these forests. So we can expect them to be more influential on the carbon cycle, for instance. Then when we look at shifts locally as well, there seems to be shifts in abundance across resource gradients and topographic gradients. This study by Davies et al. Done in Kruger here shows this very well, where you can see that every black dot here is a termite mound. And we can really see that they tend to cluster in specific areas in the environment where they like to be. So what consequences does this have for carbon cycling? And for this, I'm going to use an Australian case study to look at this a bit more. First, we looked at regional patterns in how termites change throughout the landscape. So for this, we used a precipitation gradient across the far north Queensland and northeast Australia. So from a rainforest environment with about 4,000 millimeters of rainfall to a driest savanna site with about 800 millimeters of rainfall. So we've got a good precipitation gradient from this rainforest to this savanna. So we know from a previous study by Rebecca Clement et al. In 2021 that termites tend to be more abundant and have a high species richness in these savannas. As you can see from each dot here is a termite mound, and we can see that in the two savanna sites, there tends to be much more termites than there are in other sites, such as this rainforest site. So what consequences does this have for ecosystem function and the decay rates of wood? So for this, we set up a wood block decay experiment. So we set out pine blocks out in the wild, put them in mesh bags, set them out for 4 years, and collected them every 6 months. So half the mesh bags had holes in them on the bottom, which allowed termites to access these wood blocks, and the other half didn't, which showed more of a microbial decomposition pathway. When we collected the termites, we looked at whether there was termite damage or not. So as you can see on this block, clear termite damage. There's clear galleries that the termites shows that the termites have been feeding in them. And this block is much more smushy, much more fungi-like. We can see the difference in structure in these two blocks, and then we can attribute the damage to either fungi, either termites, essentially. So to look at the decay, we first look at 2 different steps in the decay of wood. So especially for termites, first of all, the termites have to discover the wood. So for this, we looked at the proportion of blocks discovered by wood. So here's a termite going through the system, eventually finds the wood. Once it finds the wood, it starts recruiting more workers to start consuming the wood. So then we calculated the decay rate of the wood blocks, which essentially shows how fast the termites consume the wood in the system. So what did we find? So first of all, if you look at on the x-axis here, we have, so from the beginning of the experiment to the end of the experiment, and on the Y, we've got the proportion of blocks attacked by termites. We can clearly see that as the experiment progresses, we get this higher discovery of termites, which we kind of expected because as the wood blocks are out there longer, termites can find them better. But then what was really interesting is that there was about 10 times more discovery of the wood blocks in the 2 savanna sites as there were in all the other sites, showing that in these savannas, more carbon goes through these termite pathways than they do through the microbial pathways, supposedly. So then when we look at consumption rates, what did we find? So first, if we look at the overall consumption rate, so termites and fungi, we can see that there is about a similar decomposition rate across the rainforest site and the savanna site here. So this is time to 50% mass loss. So it took about a bit less than 3 years to get to this 50% mass loss threshold. Then when we separated within termites and microbes, so we've got the microbes in the green here, the termite pathway in the orange, we can see that microbial decay tends to be much slower in the savanna site than it is in the rainforest site. However, the termite decay rate seems to be pretty similar. So this compensation so the reason these 2 sites are probably quite similar in the decomposition rates is due to the higher prevalence of termites in these systems. But then when we looked at the other savanna site, we were a bit surprised and we found that actually decomposition was slower in these other savanna sites. And a reason for this might be that there was more fire in this system. So during an experiment, we had 2 fires during the experiment. So we think that maybe there's pressure from fire, but it could also be due to other things as well. So after looking at these regional patterns, throughout my PhD, I also looked at local patterns and how these can influence termites on a much slimmer level. So, and especially I was looking at resource gradients. So as we can see in this picture, there's tends to be concentrations of a lot of wood, for instance, in this system. We can see that the wood tends to concentrate in the bottom of valleys and then grass in other parts of the system. So showing these clear resource gradients. And these tend to be changing with global change, with shrub encroachment, differences in fire regime, differences in grazing. The system is constantly changing to form these mosaics of grass and wood. So try to understand how the termites respond to these changes, essentially. So we'd expect that when there's more trees, you could get more termite mounds, such as Charles et al. Had chosen in 2021 in Kenyan savannas. And when there's more grass, we could expect that there's more grass-feeding termite biomass. So do these variations in resource availability influence the role of termites in decompositions? So for this, I'll take you to a drier site in northwest New South Wales in Australia, which is a dry grassland with about 200 millimeters of rainfall a year. And it was trying to understand how these different topographies change resource availability and such. So to put in context with the other sites, we can see that termites tend to discover a very large proportion of wood in these desert ecosystems compared to the other ones. So after a year in the experiment, we found that about 70% of wood blocks were discovered in the desert compared to the savanna sites I was talking about earlier, showing how more omnipresent they can be as well in these really dry, harsh ecosystems. And then when we look at consumption rates, we see that termites really have an extremely high consumption in these systems compared to savannas and rainforests, for instance. So for this, I use both trophic cascades and topography which modify resource gradients. So in Australia, the dingo is a top predator, and it's been removed from large parts of the landscape. So where you have dingoes, you tend to have less kangaroos, which means more grass. So you could expect that there's more grass-feeding termites in the presence of dingoes. And then on the other hand, you have dingoes through mesopredator release are essentially hindering the recruitment of wood. So they're leading, you'd think there's less wood, more grass, so less wood-feeding termites and more grass-feeding termites. And then this is also put in context of the landscape. So you have these dunes which always tend to have more wood than the swales. So the dunes are really woody here, the swales are much more grassy. So on the dingo side, you tend to get more grass on the swales than the swales on the no dingo side. And then for wood, it's the opposite, where you tend to get more wood on the dunes on the no dingo side than on the dingo side. So this has been demonstrated through research in the past 5-6 years. So how does this influence termites? So we found that there was a high consumption of wood on dunes and on swales, which goes with our hypothesis that more wood leads to more termite consumption, so that was good, but we didn't find that there was an influence of the presence of dingoes on these contributions, which may be due to something else, which I'll talk about later. Then for the grass, it was a bit more complicated, where we find that there was a high consumption of grass on the dunes in the absence of dingoes, which goes totally against what we were expecting. So this is not really related to resource availability. We think that this might be due to maybe top-down predation pressure. So the dingo, by favoring small mammal insectivores or grazers, essentially put more pressure on termites as well. So there's maybe this top-down pressure which is hindering their ability of termites to contribute so much as decay, as Paul and Kate have also showed with the ants and the termites, for instance. Also, the grass feeders in our systems, in these really dry systems, have a very flexible diet, as they have to adapt to these dry conditions and can go from litter consumers to wood, to bit of grass, so they're very flexible diets. So to sum it all up, essentially we found that termites are significant components of decomposition, especially in these dry sites from savannas to deserts. But their effects tend to be quite variable, so more important for decay of wood in dry savanna than in rainforest. When you get high wood availability, you can get a higher contribution of termites to wood decay, but that does depend on other factors such as top-down predation pressures, for instance. And so to sum it all up, I'd like to acknowledge all the help I've had from amazing lab group, the Zan Lab, and all collaborators which have helped with putting this together. Thank you to all the volunteers as well, which really helped to make field work much easier and much more fun. And thank you to the NSF and the Australian Research Council for funding. Thank you.
Thanks, Baptiste. You used your time really well. So we do have a couple of minutes. If there's any questions, can you just raise your hand and we'll bring you a mic. Isak.
Thank you, Baptiste and Paul, for very interesting talks. I was wondering, in Kruger in 2015-2016, we had a severe drought, and the termites seemed more active, but it might also possibly be just that one could see their activity better. But in terms of the thermal limits that were explained and their high activity in drier places, I was wondering whether you think they also respond with insight to conditions changing and whether their role during droughts can potentially change and differ. And there's obviously a global change perspective to this question as well. So just your comments on that would be interesting. Thanks.
Yeah, that's a good question, yeah. So I think it's important to put the role of termites in contrast with the role of microbes as well. So during droughts, you tend to have termites contribute more to decay. Maybe if it's not absolutely more in some instances, they will contribute much more percentage-wise than microbes and fungi, which essentially can't function at all during these really dry, more barely function during these dry periods. So termites just have the upper end in these systems. So it's this relative contribution that really changes during droughts, I think, yeah.
Great, thanks. We still have time for one last question. I think there was one on this side here. Thanks, Tally.
Hi. I was just wondering how you adapted your experimental design to fire. So you've got pine blocks, and how did you manage that?
Sorry, how did we manage pine blocks?
You've got an experimental design where you can describe what happens in relation to fire, but your experimental system is susceptible to fire.
Yeah. The pine blocks themselves. Yeah, well, yeah. So we couldn't use a native species, unfortunately, to test these trends, as on this rainfall gradient, there's not the same species throughout these different sites. So we did use the pine blocks, which are susceptible to fire, but the fire does tend to consume a lot of other wood in these systems as well. Yeah.
Thanks, Baptiste. So we have run out of time, so thank you very much for a great talk. And any other questions, grab him a tea. Thank you. So we're still staying on termites, and our next speaker is Ellen, and the effects of long-term fire regimes on termites. Thank you, Ellen.
Thanks, everyone. So I'll be talking about my PhD research looking into termites, tree hollows, and long-term fire regimes in the tropical savannas of Australia. Now, before I start, I'd like to acknowledge the Larrakia people as the traditional owners of the land on which I live and conduct my research, and I pay my respects to their elders, past and present. So for a quick intro into Australian tropical savannas, they occur across the northern part of Australia, and they're characterised by a eucalypt-dominated canopy, a shrubby mid-story, and a grass-dominated understory. The climate is wet, dry, tropical, with an average rainfall of about 1,400 mil, and most of that falls within the wet season. So about 90% of that rain falls between November and April. Now, these savanna ecosystems are also characterised by frequent fire. The fire in these areas accounts for almost 70% of the annual fire extent in the entirety of Australia, which is pretty impressive for just that top band. This map highlights the frequency of fire in these areas. So yellow and red areas are areas of land that are burned every 1 to 2 years. And deliberate fire management is a major contributor of this high fire frequency. So this could be to reduce the risk of severe late dry season fires, for greenhouse gas abatement, so land managers can actually earn carbon credits for burning their land early in the dry season, as well as for cultural and ecological reasons. Now, a problem with this frequent burning is that we don't really understand how it affects the environment, especially in the long term. And if you want to manage your land well into the future, it's important to understand this. So for my project, I'm particularly interested in how these long-term fire regimes affect tree hollowing. So tree hollowing is really important as a habitat resource for many Australian native animals. So birds, mammals, reptiles, they can all rely on tree hollows as a place to shelter and nest. And if you don't have enough of them in the landscape, then that can negatively affect their ability to persist in an area. So in these savannas, we know that both fire and termites are major drivers of this tree hollowing. So fire, yes, it can kill the trees and knock them over, reducing the number of hollows, but minor damage to those trees by fire can actually promote the development of new hollows. And termites, well, they're the major creators of tree hollows. They eat the wood, leaving behind the hollows in the tree. And it's estimated around 84%, so every 8 out of 10 eucalypt trees that you see in the Darwin region actually have some degree of hollowing by termites, which is a lot. There's also probably something going on between fire and termites that we're not entirely certain about, but does have flow-on effects to the number of hollows in the landscape. So fire, while it might not directly kill the termites because they're generally sheltered underground, it does change the vegetation structure of the area. So this might change what termite communities it can sustain. So if you have fewer woody species, you might have fewer wood-feeding termites and fewer hollows. Going the other way, termite damage to trees can make them potentially more vulnerable to fire, more likely to fall over, and thus reduce the number of hollows. So what don't we know? Well, we don't know how long-term fire regimes affect termite communities or tree hollowing. The few studies in Australia are short-term, so over a period of 8 years, and only really use the ambient fire frequency, so what naturally occurs in the landscape rather than stuff that's been consistently applied. We also don't know specifically how different termite communities affect tree hollowing. And we need to know both these things if we're to sustainably manage our fire into the future and protect that habitat for our wildlife. So to answer these questions, I've been using a long-term fire experiment and combined that with termite surveys and hollow surveys. Now, the long-term fire experiment I've been using is the Territory Wildlife Park, which is about an hour out of Darwin. It's a public tourist attraction, and you can actually see our experiment in the satellite imagery here. So it consists of 18 1-hectare plots, broken up into 3 separate blocks here. And these represent 3 replicates of 6 different fire treatments. So we have early dry season burns every year, every 2 years, 3 years, and 5 years, as well as late dry season burns every 2 years. Now the difference between these early and late dry season burns is when they occur. So the early dry season burns occur usually at the start of the dry season, that first week of June. Whereas those late dry season burns happen at the end of the dry season, usually the first week of October. And they tend to be a lot more intense because the vegetation has all dried out. We haven't had rain for months, so the fires tend to burn a lot hotter and faster. And then we also have an unburned fire treatment, and now they've been unburned for about 32 years. So in total, this experiment's been running for 19 years now, and we're actually in our 20th year this year, which is pretty special to be part of. So here's some footage of some of our beautiful tropical savanna, as well as us burning one of the experimental plots. And you can see it's quite a big operation. We have numerous light vehicles on the ground, as well as fire trucks, because we have to work with the local fire brigades to make sure that the fires are controlled and don't get out, because it is a public tourist attraction and a lot of public are nearby. The plots tend to burn a little bit differently depending on the vegetation, so dense grassy plots tend to burn a lot hotter and faster. But as you can see, it's a big operation and we run it every year. So after 19 years of burning, the vegetation structure in these plots looks quite different. So if you take the late 2 burn, so late burn every 2 years on the left here, it's quite open. There's not much of a shrub layer, and it's got a grass-dominated understory, and most of these are annual grasses. Compare this to the unburned plot on the right. It's much more closed. You have a dense shrub layer and sparse grasses with a little layer. So in each of these plots, I've done termite surveys. So this is a combination of direct searching, so getting down on your hands and knees and trying to find termites, as well as mound surveys and baiting with wood and toilet paper. So with all these methods, I can collect samples of termites and find out what species are present. And the baiting method on the top actually has the added bonus of being able to see how active the termites are in the ecosystem, and how that might vary between the different fire treatments. So this can vary from not very active at all, so barely any bait consumed, all the way to extremely active and almost all the bait completely gone. And that can happen in a matter of 3 weeks. So in terms of preliminary results, I've run an ordination on the different termite communities and species present for the different fire treatments. So points and clusters that are closer together are more similar, and those further apart are more different. So for those intermediate fire treatments, the E2s, 3s and 5s, which are the blue and green circles here, there is a lot of overlap. So there's not a lot of differences between these communities. Where we do start to see some differences are the more extreme fire treatments. So the late 2s in orange, the annual E1s in yellow, and the unburns in grey. So there is some differentiation between these communities, indicating there's some differences in the species compositions. So a fire regime accounts for about 37% of the variation in these communities, and another contributing factor is block. And block is an unintended part of the experimental design. So if we go back to the aerial image or the satellite imagery of our experiment, you can see our plots running north to south here. Unfortunately, there's also a soil moisture gradient here, so the plots in the north tend to be deeper and, shallower and drier, and the plots in the south are deeper and wetter. And so this results in some variation between the fire treatments as you go north to south. So to look at this a bit closer, I've plotted the fire activity against each of the different fire treatments. Fire activity is the accumulated fire intensity calculated for all of the burns across all of the years. So it counts for the frequency as well as how hot those fires burn. And as you go from the low frequency unburned plots on the left to the high frequency annual burns on the right, we do see an increase in how much fire activity there is. But there is also a lot of variation in some of these treatments, which can mean it's difficult to detect an effect of regime on what you're studying. So when I took a look at how termite abundance was affected, I did this against fire activity. And as fire activity increased, we saw a decline in the abundance of termite species, or termite occurrence. So how does this translate to how active they are in the ecosystem? Well, I did actually find an effect of regime on these termite activities. It's not a massive one, but it is present. So looking at the different fire treatments on the X, most of them have a similar wood and bait consumption levels. Where we do see a bit of a difference is those late to high intensity burns, which have a decline in the amount of paper and wood bait that's consumed. So I expect that this might be due to the changes in vegetation structure. So if you remember back to that image I showed you of those late burn plots, they're quite open, there's not a lot of woody vegetation. So there's probably not many wood-feeding termites there in the first place to eat that wood. But these are analyses that I'd like to run further down the track. So what I have so far is increase in fire activity is associated with a decline in termite abundance, and those L2 regimes are also associated with a decline in that paper and wood consumption. So does that mean we're going to see a decrease in the number of hollows for those situations? So to work this out, I've been using all sorts of cool technologies, so drones, sonic tomography, and LiDAR. So with the drones, I fly my drone high up into the canopy of the tree, and I look for tree hollows. And when I see one, I can zoom right in and see, A, is it actually a tree hollow? Because sometimes what looks like a hollow from the ground can just be a snapped branch. And B, is it actually available for animals to use? So sometimes these hollows can be blocked by the same termites that create them. So an example of this is on the left. This hollow is completely plugged by termite material, so animals can't actually use it. The added bonus of using a drone is I can see hollows that might not be visible from the ground, so this one's right in the V of a tree, so you wouldn't have seen it. And then if you're super lucky, you can actually see something using a hollow, but that's a pretty rare occurrence. So sonic tomography, this is a technique used by arborists to non-destructively assess the internal structure of trees. It's like doing an ECG for a tree, but using sound rather than electricity. So we attach these sensors around the circumference of the tree, and then we tap each of those sensors with a special hammer. And this computer here measures the speed of sound between each of those sensors and can generate a picture of what the inside of the tree looks like. So this is a cross-section where we took that measurement. Black and brown represents solid wood, and those pinks and blues, that's decayed or hollow areas. So this tree is actually quite hollow, but it's not really obvious from the outside. So finally, I've been using LiDAR to characterize the vegetation structure in each of the different fire treatments. So I've been using terrestrial LiDAR from a tripod and mounted on the back of a vehicle. And from this, I can generate a 3D point cloud of what the vegetation looks like in each of my plots. So this is a bird's eye view looking down at one of the plots colored by height. So blue's close to the ground, red's higher up in the canopy. And from these point clouds, I can extract individual trees, and what I aim to do next is see if there's a relationship between the different tree characteristics, such as DBH, canopy height, and canopy cover, with the occurrence or abundance of tree hollows. And if there's a strong relationship, I'm hoping to be able to use remote sensing to estimate the number of trees in the landscape. So next steps will be to link it all together, so that fire history from the wildlife park with my termite data and my tree hollow data, to see what relationships and what is going on between fire, termites and tree hollows, and how can we sustainably manage our fire to promote and protect our habitat for wildlife. So I have heaps of people I want to thank, especially my supervisors and a small army of volunteers without whom this work would not be possible. And if you have any questions or want any more information, please do get in touch or come chat to me afterwards. Thank you.
Thanks, Ellen, for a great talk. Your time is up, so if people want to grab your tea or lunch. So then we're going to move to the last speaker before tea, which is Abby, and looking at internal stem damage in Australian savannas. Thanks, Abby.
Great. Well, my name is Abby, and today I'm going to be talking to you about missing carbon in savanna trees. And so you've heard a little primer from Ellen, but we're going to talk more about this termite hollowing and how this is happening in different areas in Australia. So now more than ever, it's critically important to understand how carbon is being stored and exchanged in the Earth system. And so in terms of terrestrial carbon, tropical forests are really important, storing about half of the carbon in the terrestrial scape. And so savannas also come into the picture, and they're storing about 0.4 gigatons of carbon per year, about a third of that of what's being stored in tropical forests. And so it becomes very clear that both of these systems, and especially savannas, as I'm studying them in my research, are really important in terms of carbon storage and carbon exchange. And so I've been focusing a lot on above-ground structures and how they're storing carbon. And so two sources of above-ground carbon in the savanna and also in rainforests and other tropical systems. You have living trees that are sequestering carbon as they grow, and then also those trees will eventually die and be releasing carbon to the atmosphere. And so we have a couple of different ways that we measure how this carbon is being cycled. In above-ground structures, such as trees, we will take dBH and estimate height and use then allometries to assemble how much biomass the structure is storing. And then when we're looking to kind of the other end of the life cycle of a tree, for a dead tree, we'll measure the mass loss that Baptiste described, we'll measure respiration of different carbon gases, and this kind of fits together the carbon cycle for woody components of the above-ground system. And so what I've been focusing on in my PhD research is the inside of trees. So what happens when we're considering carbon for decomposition that's happening in the heartwood of living trees? And clearly from Ellen's little primer, we see that that is extensive in the savanna and something that we don't quite have a grasp on, especially in terms of how much carbon is being removed. And so generally, if we have a tree that we look from the outside and we don't think there's anything going on the inside, but if it turns out that it's either hollowed by termites or damaged by other microbial players, we would hypothesize that with that internal stem damage present, carbon storage should be reduced. So we're thinking that there's more carbon that's there than actually exists. And this is really important because we're trying to see how tropical forests and savanna systems are actively storing that carbon. So it might throw off our estimates a little bit. And you can see here, it's a nice hollow tree, so we'll be talking more about that. And we don't have a great understanding of how this is happening, whether it be all over the world or different environmental gradients. And so these are just a couple studies that I've been thinking a lot about and referencing and learning from in my research, and seeing how across both temperate regions, tropical regions, within the tropical regions, dry systems and wet systems, we have a couple estimates out there for how this internal damage is impacting the biomass and carbon storage in above-ground structures. And we also know, and you'll hear a lot more about later from my advisor, Amy, how across environmental gradients we are having either drier conditions, less precipitation, as shown in this left-hand picture. Termites have a higher role in that woody breakdown. And then microbial players become dominant as you move into wetter tropical systems. And so in conjunction with understanding what's already been researched and kind of preliminarily estimated about the internal damage of trees, we also want to take into consideration how wood decomposition will be changing across these environmental gradients. And so now steering into Australia, I've been working a lot in the Australian savanna, which is a very important ecosystem across the continent. So it's covering about a quarter of the country and 2 million kilometers squared of savanna. So there's plenty of trees to go measure out there. And you can also see, as the session name indicates, termites also have a really strong presence in the system, and we'll come to talk about more of that later. And this hollowing tree phenomenon is not new information. The native people of Australia have known about this and taken advantage of this phenomenon for thousands of years. And so the didgeridoo, as this man is playing in the center photo, takes advantage of the fact that termites and fire, as Ellen described, have hollowed out trees, and then they can be polished off into wonderful, culturally important musical instruments. So by no means is this a new topic of research. But we're thinking about it in different ways, because now we've switched to understanding the carbon cycle. And so, as I've mentioned, termites can have an impact on the heartwood. And so you can see that picture on the right. This is some termite decay of a cross-section. And then also microbial players, different fungi can get in there and damage the wood as well. So I've been asking this question in my dissertation research of how internal damage is altering our above-ground carbon stores and woody structures, specifically focusing on the Australian tropics. And we're going to talk today about two main projects. The first, where I looked at internal stem damage across a precipitation gradient in northern Australia, and secondly, taking advantage of a destructive harvest opportunity to trace this damage and learn even more. So along this precipitation gradient that you've seen before, two presentations ago, we went up into Queensland, northern Australia, and looked across 5 different sites that spanned a rainfall gradient. And so a bit of what this looked like, from drier sites on the left to wetter sites on the right, we transitioned from dry savanna to a rainforest in the Daintree. And so across these sites, we're seeing largely different plant communities, different decomposer communities, and we were looking at how the internal damage is changing across that gradient. And so one of the ways that we measure this is using a resistograph drill that we call the ResiDrill. And so at each of the 5 sites, we sampled tree stems to learn more about internal damage. So a total of 258 stems and 87 different species across these 5 sites. And so to give an idea of what this data looks like and how we start to interpret internal damage from the ResiDrill, you can see here we have traces that are paired with their ResiDrill outputs. And so as we go further into the tree, from left to right on the x-axis, left to right on the green arrow, we're measuring how much resistance the drill is experiencing. And so higher resistance indicates that there's more wood, more intact wood there. And you can see as we go through these pockets of damage, the ResiDrill signal drops off. So this is how we're using the ResiDrill data in order to quantify damage. And for an undamaged tree stem, resistance stays high throughout. And so in this study, what we found is across the precipitation gradient, from left to right on the x-axis, we're seeing much more damage. So dry sites had more internal stem damage in comparison to wet sites, which were usually less than 10%. And so the dry savannas are really coming out as important when we're trying to see how above-ground carbon is being impacted by this hollowing or microbial-mediated impact. And so we also started to look within the ecosystem. The dry savannas were largely dominated by trees, oftentimes in the Myrtaceae, which carried really dense wood. And these sites that were dry also had a lot of termites. And so we're starting to observe these interactions that we tested for and found significant between both damage and precipitation, and then also damage and the amount of termites that are in the system. You can see here in the photo, there's a little termite mound at the base of this Corymbia tree. And so it seems that termites may have a liking for some of the trees within the system, specifically in the dry savannas. And so this obviously made me ask more questions, and I was able to come up with more hypotheses about how is damage happening at different heights, for example. And so that kind of leads into the next study that I'll describe. The previous study that I told you about was only measuring damage at a cross-section of 1.3 meters off the ground, and I want to learn more about how that varies throughout the tree stem. So we go up, we go down, is it changing? Is that 1.3 meter measurement representative of the entire tree stem? And then when we take that into account, how does that actually impact the biomass that's being stored in trees? And so went up to the Iron Range, which is an amazing part of Australia, about 10 hours drive north of the sites I was previously describing, and we cut down a bunch of trees. So we destructively harvested 75 trees in order to trace this internal damage and answer that question of how gradients in position on the tree are affecting the amount of stem damage that's occurring. And so we sampled, as you can see in this little diagram, at the base of the tree, and then we went up into the canopy, into the terminal branches, and we're looking at how these patterns of damage were happening. And so in our 75 trees, about a third of them incurred some kind of damage, greater than 1%, so damage is present in the system. And we were also seeing tons of signs of microbial decay here, lots and lots of termites on the right. And this is just some of the images that you get to visualize what the damage actually looks like, which is usually pretty difficult to do if you're not cutting down the trees. And so we were also observing that about half of the species within our study system were susceptible to damage. And so as you look on the x-axis, you're seeing these different species, and then the y-axis is showing the internal damage that they were experiencing. And again, we see this trend where Myrtaceae trees are carrying a lot of damage, and they're oftentimes very dense wooded, going back to the previous study. So there's interesting biology happening here, interesting interactions with the environmental conditions, the trees that are there, and then also the decomposers. And so as we look at this pattern within the tree and seeing how internal stem damage varies, as you go from left to right on the x-axis, we're looking at an increase in damage, and then we're going from the base of the tree on the y up until the canopy. And so we're seeing this trend where a lot of damage seems to be concentrated towards the base of the tree. And where this little termite mound is perched at the bottom, that might make sense as maybe they're colonizing from the ground up. And so exploring this relationship further will help us understand how this scales up to the biomass level. But so far, we're estimating that the trees on average were about 6% damaged, but some trees reached up to 30% damage. So that's 30% potentially missing from what we'd estimate using allometric equations. And so we have three main takeaways from these two studies, and I'm excited to keep researching these topics and find out even more takeaways to share with you all. But firstly, we see that internal stem damage is occurring in the savanna, and it can reduce biomass between 6 and 30%. We still have to understand this further, and this is what I look forward to kind of going on with my PhD research. Secondly, internal stem damage is decreasing as you go from the base to the canopy. And this is important as we look to how this scales to the landscape level. And lastly, these interesting interactions that are happening with both tree species, the density of the wood, and then also the presence of termites in the system, these all seem to be predicting the susceptibility to internal damage, and then consequently the carbon that's being stored in trees. So now, as we shift to thinking about the world tropics, especially in savannas, because we know that this dry system has a really important impact on the internal damage of trees, there's a lot more to learn. And so we can add to this map, and hopefully we'll find more studies to describe how these patterns of damage are occurring in altering above ground carbon in trees. And yeah, so I hope to pass the residual off to future collaborators out there and learn more about how these patterns are affecting our above ground carbon storage. And so this research would not be possible without a bunch of really wonderful colleagues and friends who have helped make all of the tree chopping and tree drilling possible, and also very unique places of the world that I get to visit that I'm always very thankful for the land that I'm able to work on. And with that, I will take any questions that you might have.
Thanks, Abby. We do have time for one question. So is there somebody out there that has a question for Abby? Over the side here.
It's not, the microphone's not on.
It is on. There we are. They're a good talk. Thank you very much. So the question about storage versus release of carbon, he didn't mention or get into the nitty-gritty of that. So am I right in speculating that because the tree is hollow, there's actually more respiration of carbon happening? So this is basically little termite farts that are being released into the atmosphere? Is that the idea?
Yeah, so the other part of my PhD research is exactly that, termite farts. So it doesn't make for as good content as cutting down trees, but yes. So I think that as this hollowing is happening on the inside, whether it's termite or microbial mediated, you would expect that there would be a respiration release that's coming from that activity. And so termites can produce methane as part of the respiration, which has a different kind of greenhouse gas consequence when we're considering global change. And that's one of the things that I'm keen to learn more about, and I am actually learning about in the system. So yes, the carbon is being cycled through different pathways, and that's what's important to understand to account for carbon correctly in the system.
Thank you.
Yeah. anyone else can grab you a tea or lunch. So this is the end of this morning's session. So I'd just like to thank all the speakers from this morning. So if we can just give everybody a round of applause, please. And then please can people come back so that we can start at 11 o'clock after tea. And just a reminder, please take your mugs so that you can enjoy the tea. Thank you.
Hi, everyone. We'll be starting in about a minute or so. I am, however, missing my first presenter for this session. There we are. Okay. Then we'll be starting in about a minute. Okay, if we can all grab our seats so that we can continue with the termites. Okay, our next presenter is Amy Zahn. And she's going to be talking about Some Like It Hot and termite temperature sensibility.
Great, thanks so much. Thanks for the introduction, and thanks everybody for being here. So I'm going to tell you about some work that we did globally looking at wood decay. And there we go. And so why I think this is important to keep in mind, and you've heard this already a bit from Abby, is what an important role that wood is in terms of a carbon store and potentially a carbon release. So wood in forest stores about half of the carbon that's in the atmosphere. And as we all know sitting here, this doesn't take into account other wooded systems such as savannas. So there's quite a bit of quite a bit of carbon stored in woody systems. And so this is true, certainly while you have living forests. Here's our Mount Sorrow and Cape Tribulation. But it's also true once trees senesce, that carbon can be stored for years to millennia. And so what's really important to keep in mind is when there's changes to this system, when there's different pressures in the system that alters the rates and forms by which this carbon is being stored, such as global change. And so those perturbations to these carbon pools can be pushed based on how it's pushing, who's doing the decay agents. So who are those decay agents? So I'm going to focus today on ones that you've already heard. So the microbes, fungi, as well as bacteria are important globally as decayers. They're found across all ecosystems. But then in certain systems, especially in the tropics, insects can become important, and especially termites, as we've been talking about in this session. And so why does this matter? What are some of the differences between these biotic decayers? One of them is how they actually release the carbon from the wood. And so we know that everything respires, releasing CO2, but that only some of the fungi can break down a really hard component in wood called lignin. And so if they break it down, it gets released as CO2 into their bodies. If it doesn't get broken down, it goes into the soil as organic residues. So the pathway can be really different depending on which fungi are doing the decay. Termites also release CO2, but then as Abby was describing before, they also are like little cows releasing methane, which is a 30-fold worse greenhouse gas. So you can have very different pathways when termites start handling them. Termites don't have the ability to break down lignin, but then ones in Africa and Asia are the fungus farmers, as Paul was talking about earlier, and that fungus is a white rotter that can break down lignin. So even the termites, depending on where biogeographically you are, can have really important differences. And so as you're changing the rates and forms depending on who's doing the decay, that has really important consequences for how carbon's being released. Another way that they differ, as you heard from, we talked a little bit with Baptiste earlier, is how they actually find the wood. So you can have microbes that are latently present in wood as a tree senesces, and they can start doing decay once the tree dies. You can also have them landing as spores or colonizing as hyphae. And so you can't predict that any particular microbe is going to be in wood, but you know that that wood is riddled with lots of microbes. Termites, as we were talking about with Baptiste, are searching animals, so they have to go around and find the wood and then recruit their friends, so it's this two-step process of discovery and then decay. Another way that they differ is in terms of their sensitivity to different environmental gradients. And so I'm going to talk a bunch about that today. So in terms of precipitation, for microbial decay to happen, they base their decay on extracellular enzymes that need moisture to deliver it to the substrate. Whereas termites are known to have various adaptations that help protect them. They have their mounds, they do sheeting across the environment and things like that helps them to be less, we predict, less sensitive to moisture. Temperature is another important predictor. So one of the things I'm going to introduce is Q10s. So Q10 is something that we hear about for enzymes. For a 10-degree increase in temperature, how much faster does that enzymatic reaction occur? And so often for enzymes, they have a Q10 of 2. So a 10-fold increase, you get a doubling of the rate of that enzyme working. Microbial decay at local to regional scales usually has been shown to have a Q10 of 2, which makes sense because it's based upon these extracellular enzymes. We don't know what the Q10 is for termites, but we know that they're largely found in the tropics, as Paul showed, and so that they seem like they should be sensitive to temperature. So I'm going to talk about the study that we had published last year, where we're looking at the relative climate sensitivities of wood-dwelling microbes and wood-feeding termites around the globe. And we expect that microbes will have decay highest in warm, wet locations because of what I just described. And we expect that termite discovery and decay will be highest in warm locations, and that termites will be less sensitive to precipitation. Okay? And as Paul was saying, there's a large list of co-authors on this. We had 108 people in 6 continents and 20 countries that were involved. A number of you sitting here in this room, and if I haven't met you yet, because many of these people have not met, and we did this across COVID, come say hello, as I'd love to meet you in person. And a lot of this recruitment of people I did by word of mouth and also social media. So you can find us if you go to Twitter. You can look up these hashtags. We have somebody's dad helping him in the Alps in Italy, and here's somebody in New Zealand deploying the blocks. And so we have a total of 133 sites around the world. We have clear gaps in our distributions, but we do a pretty good job latitudinally. So we have equal representation in the north and the south hemisphere, equal representation in the tropics and the temperate zone. And what we did was we put out radiata pine that we encased in mesh and then cut holes in half of the bag, similar to what Baptiste described.
Okay, this is just us going back onto our generator. It should kick in within the next minute or two. Okay. Either way, something will come back in a minute or two. So we have stopped your time, but while we are waiting for the projector to come back on, does anyone have a question? There we are. Okay, if you've got a question, it now needs to be a short question.
Could we have the rest of your talk, please?
I follow up on your example, Paul, of blank slides. I think Abby's got some jokes for us. There we go. Awesome. Keep going. Okay. So, and just as Baptiste described, half of the bags had holes in them, so we can allow or exclude termites. And so here you can see these are Whitaker's biomes. We go from the tundra all the way up to woodlands, and these are mapped across against mean annual temperature and mean annual precipitation. So you can see that we didn't get anything in the tundra and not so much in boreal forests, but we covered most of the other biomes in the world. Okay, so to ask these questions, to look at decay, we used decay constant K, which is the negative log of the final mass divided by the initial mass divided by time. And we contrasted blocks that were actually discovered by termites versus those that were undiscovered by termites. For discovery, we looked at, of the termite treatment where we exposed them to termites, we cut holes in the bags, what percent actually included some form of evidence that termites found them. And then to look at both decay and discovery, we looked at mean annual temperature, mean annual precipitation, and their interaction term. So now let's look at what we found. So let's start with microbial decay. So here's the decay constant, higher K, faster rates of decay, mean annual temperature. And what we can see is that only mean annual temperature was a significant predictor of microbial decay, which was pretty surprising. Precipitation wasn't significant, nor the interaction term. We also see that we recovered a Q10 of 1.7, which was not significantly different to 2. So we recovered what regional studies have found is that microbial decay has approximately a doubling with a 10-fold increase in temperature. If we move to termites, again, K versus mean annual temperature, again, wood that was discovered by termites was only significantly predicted by mean annual temperature, mean annual precipitation, and the interaction were not significant. Here we recovered a Q10 of 6.9, which is, I think, about 3.7 times faster than microbial decay. We were really surprised by this super, super high Q10 and ran the numbers again and again and kept recovering 6.9. So termites are super sensitive to temperature, really surprising that they're this sensitive. If you're interested, I can talk further about this. We have some ideas, but we were really not sure why they're so sensitive. Some ideas brewing, though. Now if we turn to discovery, so more discovered wood, mean annual temperature here, what we see is that discovery was sensitive to both temperature and precipitation and the interaction. And so you have over here in blue is high mean annual precipitation, and in gold is low mean annual precipitation. And so let's start by looking at the blue. So you can see that at the wettest sites, as temperature increases, you get higher discovery rates. If we look at if we focus then on the driest sites, what we see is that there's very little discovery at the coldest sites. But as temperature starts increasing, we get this really rapid increase in discovery. And so the highest overall discovery across our system, across the globe, was these hot, dry locations, places like savannas. And so if we look at we go back to our Whitaker's biomes, what we see here, I didn't tell you what this was before, this is the amount of discovered. So the darkest circles are the places with the highest discovery. And this line here is essentially where the tropics and the subtropics are. So you have tropical rainforests, seasonal savannas and woodlands, and you have subtropical deserts. And so you can see that it's really these arid systems, the subtropical deserts and the seasonal forests and savannas, with the highest overall discovery. So if we put discovery and decay together, so here we have the decay constants, higher decay, and the different biomes. We go from the boreal forest up to our subtropical and tropical systems. And on the left panel is where it's just microbial decay, undiscovered wood, and on the right with the dashed are the box plots with the discovered wood. And so what you can see is that whenever termites discovered wood, they elevated those decay rates. But the differences were greatest in these tropical and subtropical regions, especially the subtropical deserts and the tropical seasonal forests and savannas. Okay, so here we're getting some interesting insights into the roles that termites are playing in terms of elevating rates of wood decay around the globe, as well as what microbes are doing, really confirming some existing hypotheses. But we didn't want to stop there. We wanted to take this one step further. So we're getting this really interesting sensitivity to both temperature and precipitation of termite discovery. And so we wanted to think about whether we could predict whether, under future climates, whether more of the world would be colonized, had the potential to be colonized by termites. And so what we did was we took our discovery model and we said that if you have over 50% discovery, that's high discovery here. And then we combined that with a WorldClim downscaling of CMIP6 climate models. We bracketed with a stronger model, so the SSP 5-8.5, and the weaker SSP 1-2.6. So the stronger model means that things are going to get a lot worse by the end of the century than they are now. The weaker model says that things actually are going to improve a bit. So those bracketed for us. And so when we looked at the maps of those distributions, here's the stronger and here's the weaker model. In gray are places where we have super low discovery now, less than 5%, and we expect that we'll continue with low discovery by mid-century. In the sort of bright green here, this is continuing low discovery, so between 5% and 50%. In the sort of pea green, this is currently already high discovery. Currently, there's already more than 50%. In buff color is where it's outside of the bounds of our study system, and so we didn't measure in places that are more dry or more hot or things like that. So we can't predict in those regions of the world. But then the most interesting is this orange box. And these orange places in both of these maps are showing where currently there's low discovery, and by mid-century we expect that these are habitats that'll be suitable for high discovery rate. And so you can see that in both models, the southern hemisphere, especially these places, much more of Africa and also much more of South America, will be suitable habitats for termites to discover. Okay, so overall, we're finding microbial decay is highest in warm, but not wet locations. Termite discovery and decay were highest in warm locations, but only discovery was sensitive to precipitation, although it had knock-on consequences for our decay. And that we predict a large expansion of high termite discovery by mid-century with tropicalization, with warming. And so I think thinking about where we began with thinking about those perturbations to these pools and these systems, if you have termites invading into these novel places in the world where they're not currently having high discovery, whether they're increasing their populations or expanding, these can have really important consequences. These are, as we've heard already from Paul, their biomass is really high, similar to humans and similar to cows. They have really important consequences for fertilization and bioturbation of soils. They can alter the rates and forms that carbon is leaving materials such as wood. We know a lot about the pest species, but only about 3% of termites are pest species. So there's a lot we do not know about termites. And so I would challenge you that as you're thinking about next study systems, you might consider starting to look at termites and termite mounds, especially following all of our fun talks today. And so our paper was actually picked up by First Dog in the Moon, and the First Dog in the Moon also thought that being a termite scientist was a pretty good job to do in the world. So you can follow us and follow First Dog in the Moon and become a termitologist with us. I wanted to mention that this amazing artwork is a collaboration with Donna Davis, and so she's taken parts of our systems with wood, termites, and fungi, and had lots of digital images and videos, so you can go to her website, and I'd be happy to tell you more about her. I'll stop there. These are questions, fun facts that my friend and former student Becca wrote, and then Donna put as artwork, so you can read those, and if there's any time, I will take questions. Thank you.
Thank you for that interesting talk. We have time for one question.
Thanks, Amy. That was wonderful. I was also kind of mind blown by the Q10 of 6.7. But as far as I understand, a Q10 is more looking at the physiological constraints within a community, and you were looking across sites. So there's other things constraining termites. So do you have any insights about how the Q10 changes within a site? That's a more realistic way to estimate a Q10. So high temperature, high precipitation with the same termite community.
Yeah, Sally, thanks for that question. Yeah, and I should say these are effective Q10s, and so it's really measuring the process of decay as opposed to the actual enzymes themselves. We have played around in cans. We had some termite mounds in mesocosms, and we played around with their Q10. I can't remember off the top of my head what that number was. I don't remember us saying, God, we're at 7. So something lower than that, but I'd have to go back and dig out what the actual number was. And I think we only took one mound and stepped it through the Q10. We were having trouble getting them in culture. So I agree, it'd be really interesting to have more of these actual values from mounds or from wood where the termites are in. And this can be a combination both of, say, population dynamics, the 6.9. It can be a combination of the microbes that are in their guts. It can be a combination of the enzymes themselves and their sensitivity. So when we're working at this scale, which is pretty different to looking at just those individual enzymes.
Thank you very much. Your time is now up. Our next speaker is Alan Anderson, who's trying to expand, or is actually not trying to expand, he's going to convince us that ants are, in Australia are the most.
I won't be talking about termites, but I must start off by saying just how grateful I am for them, because as Paul mentioned, their primary function is to feed ants, and that makes me very happy. So I'm going to be talking about global patterns of ant diversity, and it's a really good news story for savannas as they struggle to escape the massive biodiversity shadow of tropical rainforests. And so if you ask any ant specialist, where do you find peak ant diversity, they'll all say, oh, tropical rainforest, of course. And that's because In tropical rainforests, it's warm all through the year, which ants love, but also habitat complexity is extremely high. And so you don't only have the normal ants that just run around on the ground that dominate all other biomes, you also have an extremely diverse assemblage of these specialist cryptobiotic species that live entirely in soil and litter, as well as an extremely diverse assemblage of specialist arboreal species. And so you really have 3 separate communities stacked on top of each other. And if you look at the most recent map of global patterns of ant diversity, you'll see, yes, that's where the biggest, the brightest reddest, which is the most diverse faunas are, Southeast Asia. This is the Atlantic forest of eastern and southern Brazil, and more especially Amazonia. That's where peak ant diversity is, because there's 2,000 species there, a lot of species. If you look in savannas, you'll see that diversity can be high. So these are the Brazilian savannas, 700 species, not as much as Amazonia, but still a lot. In Africa, southern Africa, lots of red there. We actually have little idea about what the total fauna is, but it's probably something like the Cerrados. But then if you look at Australia, can't see any red in monsoonal Australia, which is our semi-arid tropics. So how am I claiming that monsoonal Australia is the world's centre for ant diversity based on this map? And the reason is, this map is considering described species only. And the level of undescribed species in Australia is phenomenal and in fact unbelievable. And warning, you'll struggle to believe the level of undescribed diversity in Australia. And we're only getting a sort of an idea of this now after having assembled virtually all ants collected in Australia over the last 40 years into one place and done some very extensive barcoding of them, is giving us a now greater appreciation of the unbelievable diversity of ants in monsoonal Australia. And I'll just give you a little primer of what we're talking about in terms of the levels of undescribed diversity. So this is a species of ant, Tetramorium striolatum. We know that it actually represents many species, not just one, but how many species really are in there. And so what I've done is barcoded a lot of them, and I'm just starting off with one branch of the genetic tree of this species that has 9 species in that branch. And this gives you some idea of the morphological variation. But the next slide is the scary one, where I put that branch in the context of the full genetic tree of this species. And this is what it looks like. So there's that branch up there. You get the idea. So we're not just talking about, a few undescribed species. We are talking about hundreds and hundreds of undescribed species. I first put out an estimate for the total ant fauna of monsoonal Australia back in 2000, and I came up with a figure of 1,500, which was an awful lot and met a fair bit of scepticism, because that is what you're saying, nearly as many as Amazonia and much more than Cerrado. And so then the next attempt was putting together all the specimens from the top end of the Northern Territory. So this is the northern third of the Northern Territory up here, we call the top end. We got the collection in Darwin and put them all together, and there were 900 species in there, most undescribed. And more than 60% are known from nowhere else. And we extrapolated that might mean that there's probably 1,200 species, given the limited sampling in the top end, just in the top end. And so that if you look at where the top end fits geographically with the rest of the monsoonal, our savanna zone, that would think, well, that seems to suggest there are many thousands of species in monsoonal Australia. So how reliable are these? Because this is before we did all our DNA barcoding to get some genetic evidence. And so I'm going to go through 5 case studies of single species to show you the level of actual diversity that each of these single species contain. And so the first one is Melophrus rufiniger. It was only described in the last 10 years as a species that occurs all through mainland Australia. It's a polymorphic species, so it's minor workers and major workers. And in my 2000 estimate of 1,500 species across the whole monsoonal zone, I estimated there were 10 species of that species group. But the updated 2016 analysis of just the top end, there were 11 species in the top end. So what do the genetics say? Well, here's a snapshot just in the top end. There are more than 30 species just in the top end, compared with what I thought was 11 back in 2016. And then in another region south of the top end, there's another 18 species, with only 3 shared with the top end. And so I estimated that that species actually represents around 100 species across the whole of monsoonal Australia, compared with what I thought back in 2000 when I said the fauna was 1,500 in total, only 10. And the next one is this beautiful species, Tetramorium spininode, again known as a single species. In 2000, I estimated there are actually 8 species, and in 2016, 13. But the genetics are telling us again that this is vast underestimate. So just in the top end, we have 32, and in the whole of the monsoonal zone, likely around 100. And here's another one. This is back to Tetramorium striolatum, the species I was giving you an introduction to before. I'll just say, so in 2000 I said there were 15 species across the whole of monsoonal zone. In 2016 I said, well, there were 20 species in the top end alone. And the reality is that there's 50 or more species in the top end. In that Sturt Plateau area, there's another 50 species and only 10 are shared. And I reckon that there are more than 150 species in the monsoonal zone, compared with what I thought in 2000 as 15. And another one. So Monomorium fieldi, again, 20 species I thought in 2000, 15 I thought in 2016. You look at the genetic evidence and have another look at the morphology. Here's 10 species from a single site. It's a Territory Wildlife Park site that Ellen talked about with the fire experiment. And even in a single 10 metre by 10 metre plot, 6 species within 6 of those species in that plot. So how many does that mean when that species is found throughout Australia? What does it mean? It means there's just unbelievable bucket loads of these things. And so I said 15 in the top end. Well, there's more than 50. I said 20 in 2000. I said 20 across the whole of the monsoonal zone. Well, there's more than 200 species. And finally, this group represented by 2 species, and again, similar sorts of figures, 42 species just in the Sturt Plateau, and 63 species in the top end. I thought there were 13. There's likely more than 250 species across monsoonal Australia, not the 20 I estimated in 2000. And so if you put these all together, these are the 5 case study species I've talked about, the number of described species in monsoonal Australia. And this is what I estimated in the top end in 2016, and these are the actual figures. This is what I estimated in 2000 when I thought that there might be 1,500 species in monsoonal Australia, and this is the actual figure. So we're talking about an order of magnitude greater than my figure of 1,500 back in 2000, which then was quite outrageous. And so if you extrapolate that, you come up with, well, so it's 10 times as many. Does that mean there's 15,000 species of ants in monsoonal Australia? Because that's how many species there are in the world, known in the world. Amazonia's only got 2,000 species. So it's clear that we have got many thousands of species in monsoonal Australia. Now, one important question is, well, Alan, you've done so much barcoding. If you went and barcoded other places, would you get a similar degree of unrecognised diversity? And the answer is no. So here's an example from a study that did just that in the Atlantic forest of Brazil. So that's exceptionally high diversity of ants there. And they did lots of barcoding and came and asked that question: how many additional species does the barcoding reveal? And the answer is, well, just not many, actually. Not an order of magnitude more than what we recognise. So we don't know exactly how many species there are in monsoonal Australia, and I said if you extrapolate from these figures, it's indicating 15,000, which just is too ridiculous to believe, but it's clearly many thousands. And so it is clear that the conclusion is, Amazonia, sorry, you have to move over. That the true global centre of ant diversity is in a tropical savanna landscape. It's in monsoonal Australia. Thank you.
Okay, thank you very much for that, Alan. We actually have time for 2 quick questions, or 1 long one. Okay, there and there.
Thank you. Very interesting presentation. Do you have any idea how your region compares to other savanna areas that might also be poorly described, such as savannas in South Africa?
So the answer is yes. And so less so in South Africa, but certainly in Brazil. And the Cerrado fauna is known very well. And that figure of 700 species is pretty robust. One of the interesting things is that site-level diversity in the Brazilian Cerrado is comparable to that in Australia, but the turnover across space is very, very low. And so you can go from 150 species within 1 hectare to only 700 species in the whole massive biome of Cerrado. Here in southern Africa, site-level species is nowhere near as high, but compared with Cerrado, the spatial... I've got to say, we just don't have the data in southern Africa, we don't. But my experience is that the spatial turnover is much higher than in Brazil. And in terms of what that means for the whole regional fauna, say, of the savannas of southern Africa, it's really hard to know. But nothing like the thousands that we're talking about in monsoonal Australia.
Hear me? Great talk, yeah. I was just wondering, have you looked at functional diversity as well within the different, like all these groups that you're splitting into many species, do they tend to be functionally diverse as well, or do they all feel the same niche?
No, that's a good question, and the clear answer is absolutely no. So this diversity is within very closely related species complexes that, as far as we know, are functionally equivalent. And so the data I showed of the tree with the species from the one 10 metre by 10 metre plot, so those species, we spent a lot of time recording every single ant niche we could think of, from their morphology to their behaviour to what they eat. We just basically couldn't find any differences. So that's the remarkable thing about them, is that they seem to be, to a large extent, ecologically equivalent, yet we can have vast numbers co-occurring.
Okay, there does seem to be quite a lot of interest. You have now officially run out of time. So unfortunately, all the other questions that are waiting, please grab Alan over lunch. Okay, it is time now to move on slightly, going from pure ants down to ants, beetles, and spiders. And please excuse me for massacring your name. Tarumbera Mvavu.
That will do. Good morning. We've been working on and looking at the effects of anthropogenic disturbances on the species composition of soil invertebrates, including arthropods. over the past 5 years, I think. And the focus in this talk is on beetles, ants, and spiders. As you know, the savanna provides, supports many life forms, including arthropods, and the surface active arthropods are also quite abundant in the savanna, and they are confined to very specific habitats. For example, we have found that the species composition of many arthropods that we find on the rock outcrops here in Mpumalanga is quite different from the species composition of arthropods that we find around the rock outcrops in the savanna. The savanna has been transformed a lot. In Mpumalanga, we have subtropical fruit plantations, pastures, and residential areas are spreading. And thus, a lot of groups of arthropods are threatened because of the destruction or the modification of their habitats. This is more so for those groups that have limited dispersal abilities. And so we started looking at the effects of these disturbances on the species composition of the soil invertebrates. But my talk, as I said, is going to focus on the beetles, ants, and spiders. We carried out this study in the Barberton Nature Reserve, which is under the MTPA, and also in all the areas around the city of Mbombela where we identified disturbances, human disturbances. We did not really group the disturbance to say this is pasture or this is due to fire or frequent fires. We just lumped them up as disturbances. The Barberton Nature Reserve, which we looked at as our pristine site, is about probably 20 or so kilometers outside the Mbombela municipality area. We collected the data, our sampling was between December 2018 and March 2019. We had 15 sites in the Barberton Nature Reserve, which represented our undisturbed sites, relatively undisturbed sites. And we had 15 sites as well in the various parts in the Mbombela municipality area that were the disturbed sites. The sites were 10 by 10 meters in area, and we used pitfall sampling, where we had 24 pitfall traps within each site, arranged in 4 rows of 6. And we had 4 intervals where we would insert, say, pitfalls today, and then after 7 days we pull them out and then come back to the same site and reinsert 4 times within that study period. We then pulled the samples from each of the two categories, the disturbed savanna and the relatively undisturbed savanna, and took the samples to the laboratory where we then identified them put them into morphospecies, counting them, and then carried out our analysis. That picture there is of a student who was doing all the dirty work. We found that ants and beetles, their occurrence was greater in the disturbed habitats than in the undisturbed habitats. There were no differences in the species richness of the spiders when we compared these two categories. We also found that the abundance of ants and spiders was also higher in the disturbed habitats than the undisturbed habitats, and there was no difference in the abundance of the and the abundance of the beetles. The species composition was also different when you compared the three taxa in the two categories that we were looking at. What was most interesting was that the disturbed areas had more than 2 times the number of indicator species than the pristine savanna. Our results, we try to explain them with using the intermediate disturbance hypothesis, where it says that where we have intermediate disturbance, you tend to then get coexistence of maybe specialist and generalist species, and results in a very high species richness. And however, we also find that the response of the various taxa would be different to the various disturbances that occurred. But what we did not do, as I said earlier, was to separate the types of anthropogenic disturbances so that we could get a clearer picture of the effect of these various disturbances on the species composition of the anthropods that we are looking at. But what is clear from our studies that the disturbed areas, at least around Mpumalanga, that we sampled in, are biodiversity hotspots for arthropods. And thus, in future, we may need to look at not just the protected areas, but also look at the disturbed sites when we consider conservation of arthropods. Thank you.
Thank you very much for that talk. There is still quite a bit of time left, so is there any questions?
Yeah, thanks for the interesting talk. It would be nice, as you mentioned at the end, to investigate more what you consider, what is identified as disturbed. So it would be nice just to have some thoughts from you, like disturbance as in sort of harvesting of above ground biomass versus plowing of the soil and destroying the soil, because beetles and spiders often live in the soil. Maybe you could just expand a little on the types of disturbances that you think might have different effects on the arthropods.
Yes, so what we looked at were around the Mbombela municipality area, where those sites that were close to residential areas as disturbed, where we were coming across invasives, or where there were pastures and sites that were adjacent to subtropical plantations. So the variety of what we refer to as lumped together as anthropogenic disturbances, which is why I said at the end there that we obviously need to then tease them out so that we could then identify specific disturbances and try to look at those effects of those particular disturbances on the species composition of arthropods. But those were some of the disturbances that we came across. There's a lot of harvesting of wood, for example, harvesting of medicinal plants that we also identified in these areas that we're looking at.
Thank you very much. Final question? Okay. Thank you very much for your talk. And with that, we are moving away from the insects, and we are now moving on to the forests. So our first talk in this section is Tristan Charles Dominique, who's going to be looking at why specialists cannot invade forests.
Thank you very much. So in this talk, asking a very simple question with very difficult and complicated answer. So why the open specialist? And I'm talking about woody plants that are living in the savanna, they cannot get into forest. So for this talk, we're going to Schlosslo in Folosy. So it's another park. If you haven't been there after the conference, you don't stay in Kruger, you go straight to Schlosslo and you visit it, because it's a wonderful place, where you can find forest together with savanna, and also with these blue patches that are thicket. So it's another closed biome, but not as shady as a forest. So we did some earlier work and showed that you've got very different set of species in savanna, in forest, and in thicket. And then when you see this kind of spatial pattern, what is causing them, what are the drivers that make this entity discrete. And one of the ideas that in savanna and thicket, fire and herbivory are very important for organizing space. In the forest, it can be shade that become the constraining variable on distribution of species. So the big question is why open specialists cannot invade forest? The trivial sub-question is there a matter of cost in it? So are they doing something that is too costly to be produced in forest, and when the light is limiting, the open specialist could not grow? And the non-trivial sub-question is which cost are we talking about? So if an open specialist is too costly to go to the forest, why? So we did some transect of light with this wonderful tool that is a canopy imager, and that takes this kind of picture. And after taking the picture, it calculates the course of the sun in the picture. So it tells you how much light should be arriving at the spot where you took the picture. And we did transect of light from very open to very closed canopy, and then look at what is growing below this canopy. So it looks like that on the ground. This is a very open savanna. This could look like an encroached savanna, but this one is a thicket patch. And then once you get into forest, you have different level of shade that the eye is not really good at perceiving. But basically, when you are at the deepest part, you keep looking high to look at the plants because there's almost nothing on the ground. So it's shifted to another places. In this gradient, we noticed that the plants are spreading more and more, the canopy. And we wanted to compare what allows the plant to spread, what prevents them to spread the canopy. So we designed this measurement, the branch mass per area. So we cut 1 kilogram of branch. And this kilogram, then we extract the intercepting areas. So in it, you see leaves and stem. So they would both contribute to the cost that we are calculating. And we have to keep in mind that a plant can never produce leaves without stem. So calculating the cost only on leaves, it would miss a part of the equation. So this is some example from open specialist to thicket specialist to forest specialist. And you see the variation of area of 1 kilogram that is intercepted by these 3 species. And then we plotted the branch mass per area that we encountered on different individuals, from completely open system to completely closed system. And what you see is this drop here. So it means that probably a species with a BMA of 0.20 could not get into the forest. You can separate species on this way and say, okay, if I got BMA that is over this value, then I cannot enter thicket, I cannot enter forest. If you have a BMA that is below this value of 0.10, then you could enter forest if it's only a matter of shade. And then the question is, what influence this biomass per area? So here are the 3 species on top of each other. And then there's huge variation, and we try to understand why there's this variation. So this is what we try to explain, the ability to catch light of plants. And we're looking if there's difference between evergreen and deciduous species. So we're also looking at the way plants can make leaf thinner and then less costly. So that would be what would be expected in forest, in deep shade. Then, of course, the plants from open habitat, they also are in an open habitat because it's maintained open by something. And this something can be a large animal that is coming to eat the leaves. So many of the species in Shushluwi got spine that are very efficient against medium-sized mammal. So we're wondering, if you get spine, does it increase the BMA so you cannot get into the forest? There's some architectural parameters. So here we're looking at the growth direction of the branches and the trunk. So it's which direction the main structural stems are growing. And then we've got a lot of little adaptation to open ecosystems. So if you look in disturbed area, you find a lot of species that have more than one bird per leaf. And they use these extra birds to resprout after both clipping by herbivore or burning after fire. The species that are in fire-dominated place, they protect the bird below the bark, and we're wondering if it induces a cost or not. If they are producing below the bark, there's a bark story, so they are producing much more bark. How much is it influencing the ability of plants to grow into shade? Then we recorded the wood growth rate, so it tells you how fast the species is growing. So it separates fast species from slow-growing species. And this was expected to be one of the biggest explanations about why species can do well into forest, according to succession theory. And we recorded the wood density, assuming that if you have high wood density, it's more costly, and then you would get a balance affected between the biomass per area. So we recorded this trait for 45 species, and I will ask if we can move the little windows at the bottom of the camera. Sorry, can we move this a bit? So we recorded this trait for 45 woody species. Then we transformed the continuous data with boxcox to remove the too high influence of high or too little values, or the skewness and the kurtosis. We normalized all the data to make them comparable, and then we used the stepwise regression to see which of these variables influence the branch mass per area. So this is the output of the model. So you can first look at the intercept, and that define a base value that is right above the limit of growing into forest. So when you add something, you exclude the species from forest. When you remove something, you allow the species to get into forest. And what we could see is that some traits are not affecting the branch mass per area, but some other influence it a lot. And by a lot, I mean to a degree that would prevent the species to get into the forest. So there's one thing that we can observe here, is that if you're producing thinner leaf, it reduces the cost of the structure. That was expected, but what maybe is not that expected by people out of this room is that if you produce bark, it is as influential as the strategy of leaves. If you produce spine, not as much, but not far from as effective as the strategy of reducing the cost on leaves. The branching parameters are also very influential on this branch mass per area. So if we want to put that into a nutshell, most of the costs are actually associated to stems and not leaves. And when we have to think about how the plants are dealing with shade, we have to look at the stems first and second to the leaves. And then what makes you really fit into this open ecosystem dominated by fire or by herbivore is actually excluding you from a bit shady area. So if I have to criticize my talk, and this is what I count on you to discuss later. This is profoundly wrong to think like that, but we do not have better ways so far. So here I quantified the cost as a biomass unit, and we know it's wrong, because if you look at what you find on the table of any researcher, well-funded researcher, you find a bottle of water, a computer, and a gold ingot, and they might have the same biomass, but definitely they don't have the same cost. So for us, the cost is explained by rarity here and by the production cost of the computer. So how rare are the compounds that we find in this different structure, and that would completely influence whether it's difficult for the plant to extract it or not. And what are the production costs? And if you think about the production cost, 1 kilogram of bark, it's definitely not the same for the plant as 1 kilogram of leaf that is mostly water, when the bark is mostly lipidic secondary compounds that are very costly to produce. So this is wrong. Somehow it works, so it makes a good approximation about whether the species can go or cannot go into forest. It's a useful tool, but if there's physiologists who can tell us more about how we put apple and pear into one analysis of cost, I would love to hear it, because I have no clue about how to do the next step. And then the other thing is we use this branch mass per area, that is 1 kilogram of plant as unit of measurement. It's very nice because it integrates the different structure of the plant. But the problem that we face immediately is that we also integrate the effect of the cost mitigation strategy in the thing. So the costs that we are recording are already greatly mitigated by the plant. So I can give some example. If you look at the bark, we recorded the bark growth rate at the base of the plant, but some of the plants are protecting all the stem with bark. Some of them are only putting bark on the main trunk, and then they mitigate a lot the cost. And we don't have that as a species single value, so there's much more complexity into it. Another example would be the cost of producing leaves. And I was telling you that the plant cannot produce leaves without stem, but what they can do is they reduce the cost of the stem a lot. So here you see a long shoot, and here laterally a short shoot. And this short shoot has been growing for 11 years. So it's been producing a lot of leaves without wood, with very low cost. And actually all the acacias that you can see outside have this little short shoot. They are not long-lived, but they reproduce the short shoot. And they mitigate the cost a lot. So we've got the results, but on the way the plants play trick to reduce the cost. Finally, if you look at spinescence, you've got a lot of different situations outside. And if you look at an acacia, what they are doing is they're producing spine with a leaf, and the next year they produce a short shoot, and the next year another short shoot here. So they keep using the same spine without reproducing it. So they produce once with the cost of producing. They reuse 5-10 times, so that impacts the final cost of the structure. So a lot of mess for you to think about, and if you've got idea about how to go further, I would love to hear them, because we're at the very preliminary stage. So if you've got any question, I would be gladly answering what I can.
Thank you, Tristan. I'm definitely not going to be looking at the plant in the same way again. You've got time for one question.
Don't ask me about roots.
I don't know so much about roots. You're safe. So, Tristan, to me it seems simpler. Can't you use stoichiometry to work out a cost? So is it more a question just of turnover? Is that really the problem?
There's a big part of turnover in terms of mitigation strategies, and I think we need to start developing an ecology of time, not only what we see, but how the plants are managing cycle and reusing what they put. Instead of stoichiometry, I don't know how good a proxy it is because you can make so many things with the same, different combination of the same. I don't know, but if you've got an idea, I would love to hear it because, yeah.
Okay, thanks very much. Unfortunately, your time is up, but please, it looks like there's a lot of work that can be done. So you now know who Tristan is, so grab him at lunch. Okay, we are still in the forests. Evan Hockridge is our next speaker, and he's going to be looking at some similarities and differences.
Yes, thank you very much. Hello, everyone. So today I'm going to talk to you all about a very particular type of rainforest canopy gap that's quite unique and I think pretty important, and their perceived and hypothesized relationships to savannas in Congolese rainforests. So the clearings I'm referring to go by an indigenous name most likely, usually the term bais, and they're pretty unique in the world of global rainforest clearings. The most unique feature that is most striking and obvious to most people is their size. So bais can range upwards of 100 hectares in size and seemingly sort of just pop out in the middle of the rather dense rainforests. And we don't know very much about bais in general. They're generally understudied. One of the things we do know, however, is that they are, or at least most studies suggest, is that they are highly saline in nature and represent sort of a hotspot of resources available to a wide range of animals. And that leads us to one of the other things we know about them. And it's that essentially bais are hotspots for a wide range of biodiversity. The most obvious are plants, being giant clearings in a forest. Plants don't have a lot of other options if you are a grass or a sedge. But also to a wide range of other species. In fact, many ungulate species, particularly animals like forest buffalo, sitatunga, and bongo, have all been shown to center their entire ranges on these features. And the two most famous, I would say most famous critically endangered animals in the Congolese rainforests, being African forest elephants and western lowland gorillas, both utilize bais to a very high degree. Interestingly, these two species don't prefer the same sort of class of bais. Western lowland gorilla prefer bais that are much smaller and generally have waterlogged soils, even in the dry season, whereas elephants prefer those bais on the 50 to 100 hectare size that have drier soils in the dry season. What's interesting about both those species, and we'll come back to that concept of gorillas and elephant bais. They're usually called gorilla bais or elephant bais. What's interesting about both those species is they're both very far-ranging, and they've both been shown to travel extremely great distances to very specific bais. So this is actually a map of elephant telemetry data from Adzala-Kokwa National Park in the Republic of Congo, where I do my work. And those yellow dots in there are bais, listed as forest clearings, where elephants will travel in near straight-line distances, sometimes over 100 kilometers, to very specific clearings in very particular times. And the impact of all this animal activity in these features is that these bais are very heavily engineered by the animal species that visit them, usually due to practicing geophagy. The animals are actually eating the soil, presumably to access soil resources. It has been suggested, although mostly anecdotally, that this engineered effect, this geophagy, is what keeps these bais as clearings, keeps them as open gaps in the forest. But this has actually never been quantified ever, and there's only one study that actually all these reports come from suggesting that, and it just says it's a personal observation. We're actually working on a soil addition experiment to actually measure that change, but that effect by animals. But that's for another time. But the other thing we do know about bais is that throughout their range, which is mostly the western Congo Basin, they are beginning to be encroached by the forest ecosystem. We don't know the full extent. We are also working on a study to measure encroachment in bais using spaceborne radar satellites, as well as to actually map where bais are. We don't have a good sense of where these ecosystems are located. But this sort of, knowing that they're important and that they're being encroached has sort of led various organizations managing parks in this part of the world to realize that maybe these ecosystems are under threat and need to be conserved. And a big problem about trying to conserve bais is we don't understand part of the story, and the part we don't understand is where they come from in the first place. We don't know if more bais will be made as systems get encroached. We don't know the trajectory that essentially that they're on. But by far the most common hypothesized origin for these features is actually savannas. And the story basically goes that Central Africa for millennia has been in this essentially period of contraction and expansion of savannas and forests. Currently, we're in an era where savannas are shrinking in Central Africa. And the idea is that as these savannas shrink, animals still need resources in the savannas. Both gorillas and forest elephants actually use savannas in rainforest-savanna mosaics in Central Africa. And effectively, the density of animals per unit area of savanna increases as these land areas change. And effectively, at some point, equilibrium is met where these gaps are kept open. So one idea is that bais are quite literally savannas, perhaps modified heavily by animals, but savannas. And you usually hear that associated with a particular type of savanna that's common in Central Africa called a dembe. And dembes effectively are wet savannas that occur at sort of the edge of forest habitats in this ecosystem. And the thought is that these environments are not encroached at the same rate, which is, again, sort of anecdotal, but it does seem that woody vegetation doesn't do very well in dembes. So perhaps bais are quite literally this wet habitat. And we actually do see in Central Africa that with the level of encroachment, it's not impossible to imagine that as these savannas shrink, they can become isolated pockets. So on the left, this is actually, again, from Odzala-Kokwa National Park, just a Google Earth image. And on the right is, again, from that satellite tool that we're working with and studies coming up in the future of encroachment. So all those areas in yellow are areas that have experienced woody encroachment, as that sensor is very sensitive to woody biomass, changes in woody biomass. So basically, effectively, the hypothesis for bioorigin, it goes something like this, one of two different paths, that effectively dry savannas or dembes shrink to a given size where animals maintain those clearings and keep them open. Generally, the thought is that this is how these very large bais form, that they are early in a successional path. We call those elephant bais due to the preferred animal. And then for one reason or another, over time, these bais continue to shrink, usually forming gorilla bais, which are also formed in clusters, and the thought is that these clusters effectively come from elephant bais. There are a few people and a few textbooks that suggest it could be the other way around, that basically these dembes, which are relatively small, get isolated, surrounded by the forest, and then animal impacts actually modify the gorilla bais to be much larger and form elephant bais. But we wanted to simply know, with some preliminary data sets, are bais savannas? Do we see similar species compositions and structures? As well as, do we see evidence of a successional pattern, as a lot of the things we're working on are kind of dependent on which of these it could be. So what we did is we actually went out and collected lots of plant data. This past summer, we deployed 700, we collected data from 700 5x5 meter plots of presence-absence data of every single species. And then you can compress that matrix down to form a ordination analysis where you can isolate groups with an NMDS. And what we found is that clearly these groups are not directly overlapping. There's not a lot of evidence that dry or dembe savannas quite literally are the same thing as elephant bais or gorilla bais. But what's also quite unique is that these two types of bais also are not overlapping almost at all. They're very distinct groups, and we can actually test that statistically with MRPP, where we find the same result, that these groups are very distinct between these ecosystems, kind of refuting, potentially at least, that they're the same ecosystem. It doesn't provide support that that's successional. This is not exactly the pattern we would expect. We can take this a step further by using similarity dendrograms, which are effectively a way of looking at how different classes, how similar the community composition are between different classes. And we find, and the way you read this graph is further down the y-axis, or at least less similar to each other. And we find, first of all, that gorilla bais are very distinct, as an example. But most interestingly is that the two bai types are actually the least similar ecosystems to each other, which is not what you would expect in a successional environment. And especially they come from either savanna type, which is not really what we see here. We took this a step further by doing an indicator species analysis on 228 species recorded in these plots, and we found that 37% of them, or 84 species, are indicator species, which is, if anyone's never done this analysis, that's a very high percentage of species. And out of those, 56 of the 84, or 2/3 exactly, are habitat specific, meaning they indicate a very particular ecosystem. What's neat about this type of analysis, though, is you can also look at differences, you can also look at indicators of groups. So for example, there were 15 species that were just essentially savanna generalists that could exist in both Dembe savannas and dry savannas, which is approximately the same number that are habitat specific. What's interesting, though, is when you look at comparisons between groups, we have very few species that overlap and indicate either dry savanna and elephant bai or Dembe savannas and elephant bais. And we see a little bit more, but a similar pattern in Dembes and gorilla bais, which is not what you would expect, again, from successional patterns unless we've only recorded very extreme end members. But what's most interesting here is there is only one bi-generalist species, strongly indicating that these environments are not grading into each other over time. It's essentially a wetland specialist, using advantage of when these environments are wet. And we also only have one species that connects the three wetter environments here, which is not what you would expect of environments that are grading into each other. We also wanted to look at utilizing or assessing structure of these environments. We used drone-based LiDAR to do this, creating three-dimensional models of all these open ecosystems, which we then took that data and compressed it into a rasterized product at 25 centimeter resolution. And then utilizing a randomized plot design, we were able to assess vegetation height at a very, very high resolution within all of these ecosystems. And what we find when we do that is that, first of all, generally, and by the way, I added a new ecosystem here, it's not to confuse people. There is Katina sequences in the savannas, so I wanted to incorporate for that. So we have upland and downslope savannas. And what we find basically is that Dembes and elephant bais, despite being compositionally very distinct, are actually structurally quite similar. But again, gorilla bais sort of being this odd additional category that's very distinct, suggesting that for animals, something like animals that may respond to vegetation structure, these environments are not that similar. When we look at essentially heterogeneity of vegetation structure, we see that gorilla bais are a very distinct group, again, highlighting how distinct, both in biodiversity and community ecology metrics and the structural metrics. We don't see a lot of similarity between the different bai groups or the savannas, which they generally are thought to come from. And another cool thing that we can do actually with this data is look for proportions of these ecosystems that are represented by bare pixels, which in these very productive ecosystems almost always mean a proportion of the area where vegetation has been removed by the animals that are present within them. And what we find, which was kind of a surprise to me, is that while it's highly variable, gorilla bais have much higher proportions where vegetation has been removed by the animals that visit them. This was somewhat of a surprise because usually the elephant bais are where these big pictures of large excavated areas come from. But it does make intrinsic sense. In the gorilla bais with wetter soils, as you step on vegetation, that vegetation gets compressed into the mud, and generally the vegetation can't necessarily grow back in that same spot. Whereas elephant bais, the soil is very reworked, but vegetation can grow in a lot of places. We do have significant difference in this group at the 0.1, when alpha equals 0.1. And I think the main reason, something that's very important when we assess this group, is actually these outliers. And the reason is in elephant bais, we see very significant modification of the environment in very localized areas, which potentially could effectively mean that the way I'm assessing animal impacts isn't the best. So if anyone has ideas on how to assess very profound but localized effects from remote sensing data, I would love to talk. So the big takeaways from this is that we don't have evidence that these environments are actually successional at all. We don't have evidence that bais actually come directly from savannas, which has huge impacts on how we think about conservation of these ecosystems, especially in terms of encroachment. As it's very possible that the rates, mechanisms, and processes of encroachment in these ecosystems could differ drastically, especially when you incorporate the effects of something like animals potentially keeping these clearings open. And very frequently in these environments, bais are generally considered as an archetype, which is probably not accurate. It's probably not accurate to call all of these clearings bais, or assume that we'll get more bais as savannas get encroached, as has been previously hypothesized. So that's all I have. I will say that Eli McGill, you can probably raise your hand back there, he has a poster on this as well on some more abiotic impacts, reasons why we think these clearings and savanna types are very distinct. But that's all I have, and thank you very much.
Thank you very much for that talk. I think it does link in quite nicely as a local example of Nikki Stevens' first talk of the morning. There is time for one question. And thank you for introducing Elijah's poster. You've saved me the job.
Thank you very much. Very interesting. Probably a naive question, but given the fact that human societies have impacted, or engineered and impacted African savannas for thousands of years, the fact that in Central or South America we've seen that forests can recolonize previously heavily used areas, and the fact that you've shown result mainly from national parks in which human societies have been mainly removed. Mm-hmm. Is there any hypothesis that humans could have been involved in the creation, maintenance, and engineering of these bays? Thank you.
Yeah, so that's a very, very good question, and one that is occasionally brought up, that humans could have been involved in their creation. And the answer is possibly, especially if, I don't think they are the sole reason, because there are areas in Congo that have bays where we don't believe there has been human settlement for extremely long periods of time. There's at least no evidence. However, there are bays, actually in Atzala-Kukwa National Park, where there is human artifacts, at least several hundred years old, mostly for mining for salts. So I think humans probably do have a very similar impact on these features, although I'm doubtful that they are the origin of these features. But it's difficult to say. There isn't another analog for these anywhere in the world. There is some talk that there is a type of clearing in Southeast Asia called saipans, which are quite rare now. They've also been heavily encroached. They're almost gone, where they are somewhat similar. So it's difficult because we just understand so little about these features. But human influence, I would imagine, does play a factor on some level. It's just a question of how much.
Thank you very much. Unfortunately, you have run out of time. So our next speaker in this session is Tamron Hamilton. So we'll hand over to you. Thank you.
Hello. Hi. Hello, everyone. My name is Tamron Hamilton, and I'll be presenting my master's work for you guys today. And we're going to start off with our alternative stable states in a woodland savanna transition zone of South Central Africa. So the first step is to first define what we're considering alternative stable states. And basically, we can have these very open types of landscapes. And those are generally where we have woody cover suppression that's facilitated by fire or herbivory. And we're going to have a high grass cover. On the other side of our spectrum, we can end up with a very closed sort of canopy. And that's where we're going to have grass suppression, fire suppression, and effectively when we do get fires in these landscapes, they can be a lot more intense and have potential to damage the woody vegetation. We can boil this down to three main interactants within our system, and that will be climate, as we're measuring as rainfall, and fire, which is one of our main consumers, and the interaction of those two with vegetation. So if we zoom in on the relationship between fire and rainfall in savannas, we actually end up with switches where the system responds to. It has a positive relationship with rainfall. At lower rainfall, we have a positive correlation with fire. And that's generally where we had those smaller, less intense fires with our generally considered grassy coverage. Or we can have that negative relationship with fire, where as we increase rainfall, we get less fire activity, and that's generally those closed canopy situations where we have, when the fire comes through, it will be greater intensity with that woody vegetation impacts. So our general project aim is to compare proxy rainfall and fire records over time and assess the ecological implications of that relationship in the past. So if we zoom in, if we have a look at our study site, we are in a little bit south of where the previous presentation was talking about. So we're Central and Southern Africa. And the major components driving that system are, well, the major rainfall contributor in this system is tempertropical troughs, which take place and occur kind of in that band. And we can have the vegetation in that region, which is recently being classified. It occurs on the border between various different types of shrublands or woodlands, and in past, lots of different past papers, that vegetation has been classified vastly differently. So to kind of look at this in terms of our history, we have proxies that we use. So we have a rainfall proxy that we generate from a baobab tree core. So the first thing you need to know is baobabs are really long-living individuals. And they're able to give us a little bit of information about rainfall through the past because they photosynthesize, and carbon is taken up during photosynthesis and basically converted into glucose, which is used for tissues and growth. And in trees, we consider that, we can measure that as tree rings, or the growth in the diameter of our tree. But they also do something special. Baobabs close their stomata under drought conditions, which we can pick up within the bark or our tree rings as an increase in the carbon-13 isotope. So basically, if we collect a tree core, like this one, we can use isotopes to interpret periods of water stress in the past. And we generated a fire proxy from a sediment core. Basically what happens is sediment accumulates in hollows or basins, and we have two major scales that we're looking at here. We can have our local charcoal, which is shown in the blue box, and those are usually our bigger pieces of charcoal that get moved short distances, and previous work has kind of considered them to be within a maximum of 5 kilometers. Or we can go regional scale, which is much smaller pieces of charcoal that can travel distances. Previous work has shown 10 to 15 kilometers, but excluded crown fires, which are those more intense ones. So potentially we can be seeing signals about fire under those intense fire conditions on much larger scales. So we have each of our cores that we sampled, and we created, used radiocarbon dating to generate age depth graphs so that we can understand, make sure that we have a nice timeline to work with. Once we did that, we had a spatial variation. We did a spatial comparison to look at its representation in north and southern regions of the southern Africa continent. And then we also did a temporal comparison with sea surface temperatures from Madagascar. So, as you can see, our blue, which is our baobab tree core, does not perfectly match any of these, but it is quite far away from, for example, our sea surface temperatures, and it occurs on the boundary between our two different spatial areas. So, at the end of that, we end up with this is what our data looks like. We have fire at regional fire, local fire, and rainfall. But it's not so easy to see relationships here. And we need to think about the fact that we're also comparing apples and oranges, and that each of these proxies can have their own errors. And basically, to get around that, we compare these proxies in time frequency space, where we can look at various time scales. So this is looking at just our rainfall data. And what's important to know is here we have a positive correlation shown in red and yellow. And on the y-axis, we are increasing our time scale that we are looking at. So at the bottom here, 2 years, and at the top, in this sort of region, we're looking at something like a 50-year time scale, where we are seeing some significant trends in rainfall. If we look at the fire proxy, this is now the comparison between regional fire on the left and local fire on the right. We can see that they have two very different activity patterns that are showing up in time. Basically, around the period 1700 is where we see that shift. So putting that into a little bit of context, basically we're shifting from local scale fire to regional scale fire around 1700, and that we have attributed to a major climatic event. Many might know it, the Little Ice Age, which was generally defined in southern Africa as being very cold and very dry. So now to kind of compare the relationship between rainfall and fire at each of these different scales, what we've done is, this is now phase differences, so this is transitions between those areas where we see a relationship. And the important take-home message here is on both graphs at around that 50-year time scale, we can see a change from positive to negative to positive correlation, shown by the pluses and minuses. So now what? What does this have to tell us? If we look at that on a time scale and we relate it back to that correlation of rainfall and fire that we saw right in the beginning, these are the kind of trends that we're seeing. And if we have to interpret that in terms of vegetation and what observational data in the recent past has shown us, we can attribute these positive and negative correlations to what our observations have shown us, and that in the earliest part of our record, this region would have been a more open type landscape with very little woody vegetation, which then transitioned into something with a more closed canopy, and followed by a transition back into our open style landscape. I have a next slide, but it's only after questions. So.
Okay, there is quite a bit of time remaining, so before we move on to your next slide, any questions?
Thanks for an interesting presentation. I don't know if I misunderstood, but from what I gathered, the change in vegetation was driven by a drought event. Is that correct? Or did I misunderstand that?
So there is that major drought event that occurs. Oh, sorry. Can I go back? Yes, so if you look along the bottom here, this one, which is our original time series of the isotopes data from the Baobab proxy, over here around 1650, that's where we have that major drought event that was replicated through the our other validation point here, which is the cold air caves in Limpopo. So yes, there was a major drought event, but it's also attributed, that specifically that relationship between rainfall and fire that we're looking at as our indicator for it happening on this sort of time scale.
Okay, so it lasted a lot longer than the drought event, is that okay? Because my question was going to be, if it was climate driven, is it really an alternative stable state? But then if, I guess, you answered my question then. Thanks.
Cool. Thanks for your question.
Any other questions?
Hi there. Very interesting that you said it was the mini ice age?
The little ice age.
Little ice age. So there's actually some suggestion that that was caused by the extirpation of indigenous North Americans and South Americans, and that then suppressed the fire because the forests were kept open by indigenous peoples burning, and then when Europeans came and killed them all, then that actually led to the decrease in temperature because there was global warming happening from the previous fires. So is that conceivably related to what you're talking about?
So can I have my slide back, please? So this is some of my master's work, and there was actually a whole additional chapter where we looked at some of the different aspects that we radiocarbon dated, and we looked at some of the older wood, which does indicate humans were impacting the burning. But to kind of link to that, around 1800 is where we start seeing increases in that African population again because removal of slaves stopped.
The idea is that the death of 90% of the population in the Americas changed the climate globally, so it caused the mini ice age in Europe. And I'm asking, is that what you're seeing here in Southern Africa? Is that possible?
As far as I understand, the dynamics happening within the southern region are influenced by what was happening in the northern hemisphere. So there is potential for global scale implications to also be driving that. But it possibly could be. We're looking on the scale of regional, so I don't have a proxy for global at the moment. Yeah.
Thank you.
Oh, no, it's fine. We don't have to worry about that one. Yeah. Thank you.
Okay, so we are now moving on to our last topic for this session. So now we're going to be looking at forest expansion and bush encroachment. And our first speaker in this topic is going to be Kate Parr, that is going to look at reforestation with restoration.
Hopefully.
Kate.
You can just look at me, yeah. Okay, great. Thank you very much. So I just want to acknowledge my co-authors on this work, or my collaborators, because it's not published yet, Nikki Stevens and Mariska Tobias. So degradation and deforestation is a widespread problem. And to tackle this challenge, the UN proclaimed this the decade for ecosystem restoration, so running from 2021 to 2030. And they came up with a unified definition, which is the process of halting and reversing degradation, resulting in improved ecosystem services and recovered biodiversity. And they also highlighted that there are 10 principles that underpin ecosystem restoration. And I'm just going to highlight 2 here. One is benefits to nature and people, and the other is addressing the causes of degradation. I just want you to try and remember those, and I'm going to come back to them towards the end of the talk. So what we see is, I'm afraid these slides were clearly devised with the wrong format, but never mind. So you're going to end up with loads of weird ones going off. That's all right, isn't it? It's the content, not what it looks like. So there seems to be an obsession with trees. As part of this decade for restoration, there are many initiatives that are promoting trees. So one that you may have heard of is the Bond Challenge, and this is a really big initiative where they want to reforest these degraded landscapes, 350 million hectares globally. Now, this is underpinned by forest landscape restoration, and this is where they're planting trees to improve ecological function and human well-being across these deforested and degraded landscapes. And key to this, guiding it, as they say, is the Atlas of Forest Landscape Restoration Opportunities, that you probably have encountered through talks from William or Nikki, produced by the IUCN and the World Resources Institute. Just zoomed in on Africa here, because that's where we are, and you can see highlighted here in green are areas that they have said are ripe for restoration. And that includes Kruger, includes the Serengeti, includes the beautiful grasslands in Gabon. And that's partly because they're focusing on forest potential, so where there's the potential for canopy cover of 10% or more. So really there's this complete obsession with increasing trees, and that degradation through this lens is seen to be remedied by putting more trees in the landscapes. But of course we know that can be a problem. We know it can cause declines in open associated biodiversity. We know that it can affect water availability in these systems. We know that key processes that we need for savannas and grasslands, such as fire and herbivory, also decline. And it will affect human livelihoods. So in particular, Africa, with a large proportion of livelihoods dependent in rural areas, there's this sort of livelihoods that are inextricably linked with this natural environment. So if that changes, things will change for people too. So what we were interested in is what is the scope for inappropriate restoration through tree planting in non-forest areas? And therefore, to what extent are non-forest systems going to be targeted for reforestation or these restoration initiatives? And we looked at this in two ways. We looked at this in terms of commitments of a major global restoration initiative, and then we looked at on-the-ground restoration projects. So there have been quite a few studies highlighting that there are lots of tree planting programs in non-forest systems, but I don't think we really have a sense of the scale of this yet. So this is what we were aiming to get a better idea of. So we focus on AFR 100. So this is one of these key initiatives, part of the Bond Challenge, but just focused on Africa. And the aim is to restore 100 million hectares of deforested and degraded land across Africa by 2030. And here's an example of one of the projects in Madagascar. So all the countries in red have signed up and committed various amounts of their land to this initiative. So this just shows some of the numbers. But a total of 132 million hectares has now been pledged. So they're going great guns. They've got more than they thought they were going to get. Well, these commitments are substantial. So first of all, they're substantial in terms of the area that individual countries are pledging. So here's some, I've just shown some of them here, and in particular some of the smaller countries. You can see Rwanda and Burundi, it's more than 70% of that country area that's been committed for restoration. It's also substantial in terms of the amount of money involved. So when we look at the financing, we're talking really vast sums of money. These figures came from the AFR 100 website, but I suspect they're quite old, and I think the figures are much greater than this now. But the issue is that there are many of these countries have committed a greater area for forest restoration than they actually have of forest area available. So what we did is we looked at these different countries, and we looked at how much forest cover they had using the WWF ecoregions, and then we could see how much they'd committed and how much they were over their commitment, so relative to how much forest they had. So if you look at the bright green countries and the ones that are hashed, these are all places, all countries have committed more than they've got forest area available. So, but what does that mean in terms of the area? Well, we had a look at some of the numbers. Right, so first of all, we thought, let's have a look at those countries that don't have any forest in them. So this is the total area committed, that 133 million hectares. Well, there are 11 countries that either don't have any forest or minute patches, so we put those together. Those are the countries there. And they contribute 63.6 million hectares. So nearly half of the area that's been committed under AFR 100 is in non-forest locations. So when I say non-forest here and throughout the rest of the talk, read that as savannas and grasslands, because that's principally what it is. Okay, so there's a bunch of countries don't have forest. They're going to be planting loads of trees. But there are also countries that do have forest that have committed a greater area than they have forest available. So 14 countries like that. So we added up the area, the area of non-forest in those ones. We get an extra 19.2 million hectares, so 14.4% of that total area committed. But of course, when we're talking about forest and restoration, there tends to be this assumption that all the forest needs restoring and it's all degraded, but that's not the case. There are some countries, particularly those that have lots of forest, where some of that forest is quite intact. So we need to take that into account. And so high integrity forest doesn't need restoring, and so that portion that had been apportioned to the forest doesn't need to go there, so it's going to have to go somewhere else, those non-forest savannas and grasslands. So we get an extra 1 million hectares when we take forest integrity into account. And then we get that total of 15.2, just looking at those forest bits. And a total altogether, when we add that all together, of 83.8 million hectares, or 63% of the area committed across those AFR 100 countries is in savannas and grasslands, not in forest. So this is a lot of trees that are going to be going into these systems that we're very familiar with. What does 83.8 million hectares look like? I didn't know, so I was kind of Googling, what country is it like? It's the area the size of Namibia. That's a really big area. But if we look across all of those countries, AFR 100 countries, which is actually most of sub-Saharan Africa, and we add up how much degraded forest occurs across those countries, we get 227 million hectares. Currently, with what's happening, they're only restoring 49 million hectares, so only about a fifth of the potential forest that actually needs restoring, the degraded forest. If we took the 139 million that has been committed and said, okay, we'll just actually do all that in the degraded forest where we need to, then we could actually restore nearly 60% of degraded forest across Africa. I think that's quite an impressive number, but that's not happening because the trees are not going in the right place, basically, and the right trees as well. Well, should we panic? Because it all sounds really bad. It sounds like we're going to get lots of trees all over the place. Well, we went and had a look at the reforestation app database produced by Monga Bay of reforestation and restoration projects to have a look in these countries at actually what's happening, how much is going on. Well, we found that more than 60% of the projects in the AFR 100 countries in the database are in savannas and grasslands, and all but one focused on increasing trees, and that other one focused on grass in Kenya. Of all of those projects in the database, 85% of the projects involve tree seedling planting or agroforestry, and 50% of the savanna projects involved agroforestry. Almost 60% of those projects use non-native species, such as grevillea, because it's fast-growing. So here's some pictures of agroforestry in savannas. Does that look like restoration to you? If I said, here's a restored savanna, I think you'd be pretty skeptical, and I'm skeptical too. And so it's not to say agroforestry is not useful. Of course it can be useful for people. It can improve ecosystem health, improve soil fertility, reduce erosion, provide fruit trees and income for people. And some people say it improves biodiversity, although I'd say you're starting from a low baseline. But it doesn't enhance savanna biodiversity to any substantial extent, doesn't put those functions back into the savannas that we need, and you're introducing invasive species. So while human well-being may be enhanced, ecological integrity and functionality is not. So I think it's pretty disingenuous to be saying that agroforestry equates with restoration. It's a bit of greenwashing going on here. So going back to what I said about restoration in the beginning, and this was the definition, it's all about improving ecosystem services and recovering biodiversity, and these principles relating to benefits for nature and people and addressing the causes of degradation. I think it's highly questionable of whether there are benefits to nature. I think in most of these cases there probably aren't. And for people, I think we need a much more nuanced look at to what extent there are benefits versus losses. And addressing the causes of degradation, well, by putting trees in these open systems, as we know, it can cause these problems. So in fact, the addition of trees across this 60-odd% of Africa in non-forest systems is the degradation itself. So it's certainly not addressing the cause. It's creating more of a problem. But I wanted to say that this is a global problem. I gave you an example of Africa, But here's there was an article in the New York Times, and this is from a beautiful location in Brazil. And these people here are all planting trees, indigenous trees, forest trees, because they want all this area to be forest. The open areas in this system have a diversity of vascular plants ranging from 350 to 400 species per hectare. They would be gone with the increasing forest. So what can we do? Well, it's difficult, and this is again, another person, probably me again, like lots of people, going on about this, but we really do need to try and redefine forest. The problem is that the current definitions focus on tree cover, and they don't look at what's beneath the canopy. So we need to shift the emphasis away from just talking about tree cover. I know it's easy with remote sensing and grass is hard, but we need to do that. And we need to look below the canopy. We need to try and work towards a paradigm shift now. So recognizing the unique ecology of grassy systems because they require such different management and restoration. And again, this is something that lots of us have been banging on about for a while, but that's what happens with paradigm shifts. They take a really long time, and I need all of you guys to also just keep getting the message out there. And then finally, I just think we need to consider much more critically what degradation looks like in non-forested systems. Because clearly, according to the UN and the Bonn Challenge and AFR 100, degradation is remedied just with trees. So I'll leave it there. Thank you.
Thank you very much, Kate. We have time for one question. Okay, I think, Sam, your hand was up first. It was a close call, but...
Thank you very much, Kate, for that very informative talk. And I think you put some very interesting solutions in the end there. Most of those will be useless. And I can tell you why. Because what happens at these big international agreements and Bonn agreements and whatever else, in fact, evidence is the last thing that people worry about. It's really about who's lobbying, who is telling the best story. So what we have to figure out is how do we lobby and tell the story that you had on that last slide. We see it in several international agreements. CITES did it to us. Yeah. In fact, we had a complaint at CITES from one of the countries that CITES has just become a scientific place to talk about science. And that's the basis of decision making. So I think that's our challenge. Thank you very much for that talk.
Yeah, no, I agree. I think it is a huge challenge. It's massively political. And I think that as grassy system people, we need to talk more to the forest people. How we get them to listen is something else. But if we're only talking to ourselves, like I am now, preaching to the converted, that's no good. So we need to start having dialogues with other people. We need to get some of the grassland and savanna people into these positions in the IUCN and all these places to try and start changing things. Everything's just dominated by forest people. And yeah, here you go, challenge to all the PhD students.
Okay, thank you, Kate. With that challenge, we will release you. And then our final speaker before we break for lunch is Pierre Couturon, who's staying with Forest Expansions.
Thank you very much. Hi, everybody. So, not yet. For my talk, we'll address forest savanna mosaics and their wet climates with a special reference in Central Africa. So there are some meeting points with two previous presentations about BICE and also about the discussion on climate fluctuation. And so I am Pierre Couturon from the AMAP lab in Montpellier, France. So Tristan also comes from this lab. And the work I will present has been carried out with colleagues from this lab and also with colleagues in Cameroon from the University of Yaoundé 1. So when we have this kind of impressive mosaics between tropical, dense, close canopy tropical forest, and open savannas with a very low woody biomass. We think about managed landscape as in temperate countries with pastures and woodlots, but it is not managed at all. It is quite spontaneous dynamics with only fire-mediated human influences. And this kind of mosaic is a very extended ecotone in the northern hemisphere, especially around the forest blocks in West and Central Africa. And it has been witnessed in many places an ongoing forest progression, which still called for to be documented and explained in more details. So we have general questions, first about the savanna phase, which is the overarching question for the savanna conundrum, which is tree grass coexistence stable or is it transient? And how fire regimes modulate this possible stability. And regarding the wet savanna mosaics itself, forest savanna mosaic itself, we are looking for trying to assess the relative influences of the ongoing processes on the phenomenon of forest extension, and we have also to know better the pace, of forest encroachment. So here is the overall landscapes in which the study took place in central Cameroon. We have forest savanna mosaics, which is very dynamical. And we focused around the national park, which is unfortunately a paper park. But the area is sparsely populated. The park is about 1,000 square kilometers, and we have an equatorial climate with a total annual rainfall of 1,300 millimeters per year, which is quite a lot. And so, because herders or transhumant herders can use the park during each dry season, we have regular fires, which are not directly managed but used to rejuvenate the pasture. And in spite of those fires, forest is progressing. So first, previous studies, which is an asset for this talk, is the PhD thesis and related papers of Le Bienfaiteur Sagang from Cameroon and University of Yaoundé 1, which evidence from a Landsat series, historical remote sensing, who evidence the changes from forest cover in the park of about 1/3 up to now 2/3, more than 2/3. This was about 42 years ago. So through the historical series, we have been able to track forest expansion. Dark green are initial forest, and light green are new forest, younger forest, and orange is residual savannas. So this has been done through the Google Earth engine, and the scripts are freely available. We made also a lot of field work, and it was Le Bienfaiteur that did it most. We'll field plots in which both grass biomass and woody biomass were assessed, and we extended this through using LiDAR and drone altimetry, and also, so we had fire scars from Landsat. So we had also, in previous studies, some modeling framework from Valeriata in University of Yaoundé, which is a minimalistic model implementing grass woody biomass dynamics in Savanna. I will not elaborate much, but it is based on, I presented it 3 years ago here, logistic grow, asymmetric tree grass competition, interaction, grass fire feedback in a non-linear way, and woody biomass self-protection. I mean, high biomass is not experiencing fire impact as low biomass. So we got, it's fully mathematically tractable, so we got a lot of equilibria which are well known in advance for a given set of parameters. And regarding the field work, we found out that we have a large range of grass biomass above ground, and we can have huge values of 10 to 15 tons per hectare. And below a certain threshold, there was no fire recorded through Landsat, even though there was fire of very low intensity in the field. And the threshold is around the grass biomass of 3 to 4 tons per hectare. And if we look at data on fire frequency and above ground biomass of the woody component in Savanna maintained as such, we can see that we have a decreasing relationship evidencing something like the grass fire feedback with high fire frequency related to very low woody biomass of less than 30 tons per hectare, about that. And if we look at the grass biomass and the woody biomass, it's quite rare to have collocated data on both. And we can see that above 3 tons of grass per hectare, we have more or less no woody biomass above 25 to 30 tons per hectare. And vice versa. Under this threshold, this allows a very large biomass of grasses. So when applying the model with field or plausible parameters, we can have this phase diagram showing that trajectory starting for very low woody biomass is here, are going to what exists as a savanna equilibrium, which is unstable. We found it unstable under most plausible conditions. And then woody biomass tends to increase either rapidly to reach the forest state or either very slowly, but inexorably leading to forest. And so we can evidence through simulation that we have mainly rapid transition, but sometimes very low transition. Regarding this is a temporal model, so regarding the spatial dynamics, we did it very simply using the diachronic maps from Landsat archives. And we, as a neutral model, we implemented a simple patch dilation of a pre-existing forest patch. This was thought in the beginning as a straw man to be easily reversed. And in fact, it proved quite really relevant with a quite steady pace, with a quite steady pace of forest boundary movement, which is a dilation range of 6 meters per year, which is huge. And so we could compare, we have here the initial state in 2000. We will compare the observed versus simulated for 2020. So we see that globally it matches, but locally we can detect differences. Orange is remaining savannas. Here is pre-existing forest in 2000. Here is reforestation according of the pace of the model. And here are speeder or slower. And if we zoom in, we can see that this is a large but scalloped savanna patch. With this green, it is the pace of the model. With this blue, it is far speeder. And yellow, I don't see whether you see the fringes in yellow, it is slower than expected. So speeder, it means large patches of savannas which were overwhelmed, and slower, they are rather straight boundaries which are delayed by the FAR regime. So for the dynamics, under the study conditions, that it is quite congruent with the field and general experience, savannas are unstable, and the forest is monostable in most cases in most of the parameter space. We can have long transition when we start for low woody biomass, and this means that the woody biomass took a long time to increase, at that time, exponentially. For the overall, forest extensions, we have seen a pervasive boundary movement in spite of a yearly FAR regime, which is but unmanaged by herders, who tend to set fire very early in the dry season to have grass resprout. And in fact, very often fire is not propagating close to the boundary, especially if we have a complex pattern of forest. This is the beginning of the dry season, and most probably fire will not be set again in this part which did not burn. And simple models of patch dilation match observations and are useful to detect places where we have speed or slow afforestation. And this rapid pace of afforestation over 50 years raised the question of what is the history of those savannas, and I would be delighted to exchange with the previous colleagues, the colleagues of the previous talks. Thanks, Ines.
Okay, thank you for that very interesting talk, Pierre. There is time for questions, and I see there's already a hand up.
Thank you very much for that lovely talk. I'm really interested, so you find with your model under your current conditions, savannas are not stable. Is it possible with your model to apply a late dry season fire regime and see if the savannas then become stable? Because although human beings like early fires, and they tend to light early fires, in systems without humans, I would think that all the fires in the system would have been in the late dry season.
Yeah, you mean applying the model to compare what would happen with early fires and late fires?
Exactly. On the assumption that if there's fewer people in the system lighting fires, there will be more late season fires. To see if a stable savanna emerges.
Yes, that's part of what is possible with the model. The thing is to better master the parameters linked to the fire impact in both cases. I mean, recording fire intensity and recording loss of woody biomass in the two cases. So, of course, we can go through the literature but to try to have things like this. We did not do it yet, but we can do it. I think the model can help for that.
I hope you will do it. I'll be interested in the results.
Okay, thank you very much. And that is unfortunately your time. But it is lunch now, so if anyone wants to discuss this further, you have the opportunity. So it is lunchtime now. Just a reminder, lunch is at the Safari Lodge, so where our meet and greet was yesterday. And then we're back here at 2. Thank you.